Hello there, and welcome to Midnight Sleep Science. Tonight, you are invited to explore one of the most remarkable facts of your existence. Right now, as you settle into stillness, your heart is beating. It has been beating since before you were born, and it will continue without pause for every moment of your life. You do not think about it. You do not will it. Yet somewhere in the quiet darkness of your chest, a small cluster of specialized cells is firing electrical signals over and over, in a rhythm so dependable that your entire body trusts it without question. These cells have no resting state. They never stop. Unlike the other muscle cells in your heart, these pacemaker cells in your sinoatrial node cannot sit still. The moment they finish one beat, they immediately begin preparing for the next. This is not magic. It is not mystery. It is biology, intricate, elegant, and profoundly real. And tonight, you will journey through the science of how your heart keeps beating, from the electrical spark that begins each pulse to the vast network of nerves and signals that adjust your rhythm in response to your world. If you enjoy these gentle journeys, feel free to like and subscribe. But for now, let's begin. Your heart is beating right now. You feel it, perhaps, if you pay attention, a gentle pulse in your chest, steady and unhurried. But even if you cannot feel it, it is there, working in the darkness beneath your ribs, keeping you alive with a rhythm that never pauses, never hesitates. This rhythm does not come from your brain. It does not require thought or intention. It comes from a small hidden structure buried in the wall of your right atrium, a place so small you could barely see it with your naked eye, yet so essential that without it, everything stops. This structure is called the sinoatrial node, and it is the natural pacemaker of your heart. It is an oval-shaped cluster of specialized cells, roughly 15 millimeters long, 3 millimeters wide, and just 1 millimeter thick, about the size of a grain of rice. It sits in the upper right chamber of your heart, tucked just below the entrance of the superior vena cava, the large vein that returns blood from your head and arms back to your heart. This location is not random. It is positioned precisely where it needs to be, in a region called the sinus venerum, from which the node takes its name, sino, meaning sinus, and atrial, meaning atrium. The cells here are different from the rest of your heart. They are smaller, paler, and less equipped for contraction than the muscle cells that surround them. Where typical heart muscle cells are built for strength and force, sinoatrial node cells are built for something else entirely, spontaneity. They are self-excitable. They do not wait for a signal. They do not need permission. They simply begin over and over generating electrical impulses on their own, in a cycle that repeats roughly once per second, every second for your entire life. You might wonder how this is possible. How can a cell generate electricity without being triggered by something external? The answer lies in the way these cells are built, in a unique arrangement of ion channels embedded in their membranes. These channels are proteins, tiny gates that allow charged particles, ions, to move in and out of the cell. And it is the movement of these ions, flowing across the cell membrane, that creates the electrical signal we call an action potential. Most cells in your body have a stable resting potential. They sit quietly at a negative voltage, around negative 70 millivolts, until something comes along and disturbs them. But the pacemaker cells of the sinoatrial node do not have a true resting state. The moment they repolarize after one action potential, they immediately begin drifting upward again, slowly increasing their voltage in a process called the pacemaker potential. This slow, steady rise is what makes your heart beat automatically, without any need for external input. The pacemaker potential begins when the cell is at its most negative voltage, right after it has just fired an action potential and repolarized. At this moment, the membrane is hyperpolarized, very negative, and this hyperpolarization triggers something unusual. Most ion channels open when the membrane becomes more positive, but in the sinoatrial node, there is a special type of channel that does the opposite. It opens when the membrane becomes more negative. These are called hyperpolarization-activated cyclic nucleotide gated channels, or HCN channels for short, And the current they carry is so unusual that scientists gave it a unique name, the funny current. The funny current is funny because it defies normal expectations. When the HCN channels open in response to hyperpolarization, they allow sodium ions and some potassium ions to flow into the cell. Sodium carries a positive charge, and as it enters, it makes the inside of the cell gradually more positive. This slow influx of positive charge is what initiates the pacemaker potential, the gentle upward drift in voltage that will eventually reach threshold and trigger the next heartbeat. But the funny current is not the only mechanism at work here. There is another process happening inside the cell, one that involves calcium. Deep within the pacemaker cells, there is a structure called the sarcoplasmic reticulum, a storage compartment filled with calcium ions. Even without any external signal, this internal store spontaneously releases tiny bursts of calcium into the cell's interior, events scientists call calcium sparks. These sparks of calcium activate another protein in the membrane called the sodium-calcium exchanger. This exchanger does exactly what its name suggests. It removes one calcium ion from the cell and brings in 3 sodium ions to replace it. The result is a net gain of positive charge inside the cell, which further drives the pacemaker potential upward. This mechanism is sometimes called the calcium clock, a second timekeeping system working alongside the funny current. As the membrane potential continues to rise, driven by both the funny current and the calcium clock, it eventually reaches a point where another set of channels begins to open. These are calcium channels, and they come in 2 types, T-type and L-type. The T-type channels open first, allowing a trickle of calcium to enter the cell, which pushes the voltage even higher. Then the L-type channels open, more slowly but with greater effect, and calcium floods into the cell. This influx of calcium is what finally brings the membrane potential to threshold, triggering the rapid upstroke of the action potential, the electrical spark that will spread through your heart and cause it to contract. And then just as quickly, the cell repolarizes. Potassium channels open, allowing potassium ions to flow out of the cell, carrying positive charge away and bringing the membrane potential back down. The action potential is over. But the cycle does not stop. As soon as repolarization is complete, the pacemaker potential begins again, and the whole process repeats. This is the fundamental rhythm of your life. It is not a metaphor. It is not symbolic. It is real, happening right now in a tiny cluster of cells no bigger than a grain of rice, firing over and over with a reliability that is almost unimaginable. The electrical impulse that begins in the sinoatrial node does not stay there. The moment those pacemaker cells fire, the signal begins to spread, moving outward like a ripple across water, reaching into the surrounding tissue of your right atrium. This is not a wire carrying electricity. There are no cables, no insulation, no circuits in the way you might imagine them. Instead, the signal moves from cell to cell, passing through living tissue in a way that is both elegant and remarkably efficient. The cells of your heart are connected to one another through specialized structures called gap junctions, found at the ends of each cell where they meet their neighbors. These junctions are not solid barriers. They are tiny channels made of proteins called connexins that allow ions and small molecules to flow directly from one cell's interior into the next. When the pacemaker cells of the sinoatrial node fire, the positive ions that flood into those cells do not remain confined. They spill through the gap junctions into the adjacent cells, raising the voltage in those cells as well. When the neighboring cells reach threshold, they fire their own action potentials, and the cycle continues, spreading the wave of depolarization outward in all directions. This is why the heart is sometimes called a functional syncytium. It is not a single continuous cell, but it behaves almost as if it were. The electrical impulse propagates freely between cells in every direction, allowing the myocardium, the muscular tissue of the heart, to function as a single, unified contractile unit. This coordination is what allows your atria to squeeze in a synchronized, efficient manner, pushing blood downward into the ventricles below. The impulse spreads throughout your right atrium first, because that is where the sinoatrial node is located. But it does not stop there. A specialized bundle of fibers called Bachmann's bundle carries the signal across to your left atrium, ensuring that both chambers contract at nearly the same time. This symmetry is essential. If one atrium contracted while the other remained still, blood would not flow evenly, and your heart's efficiency would be compromised. As the signal spreads through the atria, it also travels along specialized pathways known as internodal tracts. These are routes of faster conducting tissue that run from the sinoatrial node down to another critical structure, the atrioventricular node, or AV node. There are three main internodal pathways in the right atrium, each named for the scientist who discovered them: the anterior, middle, and posterior internodal tracts. These pathways do not carry the signal in isolation. The entire atrial myocardium is depolarizing at the same time, but they provide a faster route, ensuring that the AV node receives the signal quickly and reliably. The cells that make up these internodal pathways are different from ordinary atrial muscle cells. They have characteristics similar to Purkinje fibers, the specialized conduction cells found deeper in the heart, which means they can conduct electrical signals much more rapidly than typical muscle tissue. This speed is important. The faster the signal reaches the AV node, the sooner the next phase of the heartbeat can begin. You might wonder why your heart needs these specialized pathways at all. Why not simply let the signal spread naturally through the atrial muscle, cell by cell, until it reaches the AV node on its own? The answer lies in timing. Your heart is a pump, and like any pump, it depends on precise coordination. From the moment the sinoatrial node fires to the moment both atria have fully contracted takes only about 80 to 100 milliseconds, less than a tenth of a second. This brief burst of activity is visible on an electrocardiogram as the P wave, a small rounded deflection that represents the electrical depolarization of the atria. But even as the atria are contracting, the signal is already moving onward. It is traveling through specialized pathways within the right atrium, routes of faster conducting tissue that connect the sinoatrial node to the next critical structure in the heart's conduction system. These pathways are called internodal tracts, and there are three of them: the anterior, middle, and posterior tracts, each named for the anatomist who first described them. The internodal tracts are not separate wires or cables. They are regions of atrial tissue where the cells are arranged in a way that allows electrical signals to travel more quickly than through ordinary atrial muscle. These cells have characteristics similar to Purkinje fibers, the specialized conduction cells found deeper in the heart, which means they have more gap junctions and fewer internal organelles that might slow conduction. The result is that the signal can race through these pathways faster than it spreads through the surrounding atrial wall. Why does your heart need these faster pathways? The answer lies in timing. The atria must finish contracting before the ventricles begin. If the ventricles started to squeeze while the atria were still filling them with blood, the coordination would be lost and the heart's efficiency would drop. The faster the signal can travel from the sinoatrial node to the next checkpoint in the conduction system, the more precisely the heart can control the timing of contraction. That next checkpoint is the atrioventricular node, a small cluster of specialized cells located on the other side of the right atrium, near the border between the atrium and the ventricle. The internodal tracts converge on this node, delivering the electrical signal to it quickly and reliably. The atrioventricular node sits in a specific anatomical region called the triangle of Koch, a triangular area defined by three landmarks: the opening of the coronary sinus, the base of the septal leaflet of the tricuspid valve, and a fibrous band called the tendon of Tadaro. This anatomical location is not accidental. The atrioventricular node sits at a critical junction, the only normal pathway through which electrical signals can travel from the atria to the ventricles. Between the atria and ventricles lies a dense barrier of fibrous tissue called the cardiac skeleton, a non-conductive framework that provides structural support for the heart valves and separates the electrical systems of the upper and lower chambers. This fibrous barrier is essential because it prevents the electrical impulse from spreading chaotically from the atria directly into the ventricles. Without this insulation, the coordinated timing of your heartbeat would be impossible. The only break in this barrier is where the atrioventricular node connects to the bundle of his a specialized conducting pathway that will carry the signal downward into the ventricles. This anatomical arrangement makes the AV node a gatekeeper, the sole point of passage for electrical signals traveling from the upper chambers to the lower ones. Every heartbeat must pass through this tiny structure, roughly 1 millimeter by 3 millimeters by 5 millimeters in size, no bigger than a grain of rice. The cells of the atrioventricular node are different from the atrial muscle cells that surround them. They are smaller, more densely packed, and they have fewer gap junctions connecting them to their neighbors. This reduction in gap junctions is deliberate, a design feature that serves a critical purpose. It slows down the conduction of the electrical signal. Where the impulse travels through ordinary atrial muscle at a speed of about half a meter per second, it slows to just 5 centimeters per second as it passes through the AV node. This tenfold reduction in speed creates a delay of approximately 90 milliseconds, a brief pause that is absolutely essential to the proper functioning of your heart. Why does your heart need this delay? The answer lies in the mechanics of pumping. Your atria are thin-walled chambers designed to receive blood and pass it downward into the ventricles. They do not generate tremendous force when they contract, but they do not need to. Their job is simply to top off the ventricles, adding the final 20 to 30% of blood volume that makes ventricular filling complete. But for this transfer to happen efficiently, the atria must contract while the ventricles are still relaxed, while the valves between them are still open. If the ventricles began contracting at the same moment the atria did, the valves would slam shut immediately, and the atria would be squeezing against closed doors, unable to push their blood forward. The 90 millisecond delay at the atrioventricular node ensures that the atria have time to finish their contraction and empty their contents into the ventricles before the ventricles receive the signal to contract. This brief pause is the difference between an efficient pump and a chaotic, ineffective one. The delay is visible on an electrocardiogram as a flat segment between the P wave and the QRS complex, the segment that represents ventricular depolarization. This flat interval, called the PR segment, corresponds to the time the signal spends traveling slowly through the AV node. It is a quiet moment, electrically speaking, but one of profound importance. The atrioventricular node has another remarkable property. It can conduct signals through two different pathways. One pathway conducts slowly but has a shorter refractory period, meaning it can recover quickly and be ready to conduct another signal. The other pathway conducts more rapidly but has a longer refractory period, meaning it takes more time to reset after each impulse. Under normal circumstances, both pathways contribute to conduction, and the signal passes through the node in an orderly, predictable way. But the AV node also has a protective function. It exhibits a property called decremental conduction, which means that the more frequently it is stimulated, the slower it conducts. This might seem like a flaw, but it is actually a safeguard. If the atria were to suddenly begin firing at an abnormally rapid rate, as happens in conditions like atrial fibrillation or atrial flutter, where the atria can depolarize hundreds of times per minute, the AV node would slow down conduction in response, preventing the ventricles from being driven at dangerously high rates. This filtering function protects your ventricles from electrical chaos, allowing them to maintain a more sustainable rhythm even when the atria are misfiring. The atrioventricular node also has its own intrinsic ability to generate action potentials, just like the sinoatrial node. If the SA node were to fail, if it stopped firing, or if the signal from it were somehow blocked before reaching the AV node, the AV node would take over as the pacemaker of the heart. It would fire at its own intrinsic rate, which is slower than the SA node, about 40 to 60 beats per minute instead of 60 to 100. But it would still keep the heart beating, still maintain circulation, still sustain life. This redundancy is one of the heart's many fail-safes, a backup system built into the very architecture of the cardiac conduction system. The blood supply to the atrioventricular node comes from a small artery called the AV nodal artery. In most people, this artery branches from the right coronary artery, though in some individuals it arises from the left circumflex artery instead. This blood supply is critical because the AV node depends on a constant flow of oxygen and nutrients to maintain its function. If this artery becomes blocked, as can happen during a heart attack, The AV node may be damaged, leading to conduction abnormalities and potentially life-threatening heart block. The signal has now arrived at the atrioventricular node. It has traveled from the sinoatrial node, spread through both atria, triggered coordinated atrial contraction, and reached the gateway to the ventricles. The delay begins a 90-millisecond pause that allows the heart to coordinate its rhythm with precision. And as the signal waits, held briefly in this small cluster of cells, the next phase of the journey prepares to unfold. The signal emerges from the atrioventricular node and enters a narrow corridor of specialized tissue called the bundle of His. This bundle, named after the Swiss cardiologist Wilhelm His Jr., who discovered it in 1893, is cord-like structure about 20 millimeters long and 4 millimeters in diameter, extending from the AV node into the upper part of the interventricular septum, the muscular wall that divides your left and right ventricles. This is the only pathway through which the electrical impulse can travel from the atria to the ventricles under normal circumstances. The bundle of His is surrounded by collagenous tissue, a dense fibrous material that forms part of the cardiac skeleton. This insulation is critical because it prevents the electrical signal from leaking out prematurely into the surrounding ventricular muscle. The signal must travel through the bundle in an orderly fashion, following a specific route that will ensure the ventricles contract in the most efficient pattern possible. The bundle of His is composed of specialized muscle cells unlike ordinary ventricular muscle. These cells are wider and conduct electrical impulses much more rapidly than typical heart muscle, allowing the signal to race through the septum with remarkable speed. The entire journey through the bundle of His takes only 30 to 40 milliseconds, a fraction of the time the signal spent waiting in the AV node. This rapid conduction is essential because the ventricles are much larger than the atria, and they need to contract in a coordinated, synchronized manner to pump blood effectively. As the bundle travels down the interventricular septum, it remains mostly on the left side of the wall, running just beneath the endocardium, the smooth inner lining of the heart. The bundle receives its blood supply from two sources, the AV nodal artery and the septal perforator branches of the left anterior descending artery. This dual blood supply provides redundancy, ensuring that the bundle continues to function even if one of these arteries becomes partially blocked. The bundle of His does not remain a single pathway for long. At a point called the interventricular crest, roughly at the top of the muscular part of the septum, it divides into two branches, the right bundle branch and the left bundle branch. This bifurcation is a critical moment in the heart's conduction system because it marks the point where the signal begins to spread separately into the two ventricles. The right bundle branch splits off at an obtuse angle, initially traveling through the muscle of the septum before becoming more superficial in its lower portion. It runs down the right side of the septum within a structure called the moderator band, a muscular bridge that crosses the right ventricular cavity and connects to the anterior papillary muscle on the ventricular wall. This pathway ensures that the electrical signal reaches the right ventricle quickly and efficiently. The left bundle branch is larger than the right, carrying most of the fibers from the bundle of His. It emerges from the septum and travels down the left side, initially running superficially beneath the endocardium before diving slightly deeper into the muscle. The left bundle branch further divides into two fascicles, the left anterior fascicle and the left posterior fascicle. These two pathways spread the signal throughout the larger, more muscular left ventricle, which must generate far more force than the right ventricle, because it pumps blood to your entire body, not just to your lungs. Both bundle branches give rise to a vast network of thin, thread-like fibers called Purkinje fibers, named after the Czech anatomist Jan Evangelista Purkin, who discovered them in 1839. These fibers are the final stage of the heart's specialized conduction system. They spread out like the branches of a tree, extending from the septum outward across the inner walls of both ventricles, reaching deep into the muscular tissue. The Purkinje fibers are the fastest conducting cells in the entire heart, capable of transmitting electrical impulses at speeds up to 4 meters per second. This extraordinary speed allows the signal to reach all parts of both ventricles almost simultaneously, ensuring that the entire ventricular muscle mass contracts in a coordinated, synchronized manner. The fibers penetrate from the inner surface of the ventricles, the endocardium, toward the outer surface, the epicardium, triggering contraction from the inside out. This pattern of conduction creates a specific sequence of ventricular contraction. The septum depolarizes first, then the signal spreads to the apex, the pointed bottom of the heart, and finally moves upward toward the base. This bottom-to-top pattern is essential because it ensures that blood is squeezed upward and out through the valves at the top of the ventricles, rather than being trapped at the bottom. The entire process, from the moment the signal leaves the AV node to the moment the ventricular muscle begins to contract, takes only about 30 to 40 milliseconds. On an electrocardiogram, this rapid spread of electricity through the ventricles appears as the QRS complex, a sharp, distinctive waveform that represents ventricular depolarization. The QRS complex is much larger and more dramatic than the P wave that represented atrial depolarization, reflecting the fact that the ventricles are much larger, more muscular, and generate far more electrical activity than the atria. The signal has now completed its journey through the conduction system. It began in the sinoatrial node, spread through the atria, paused at the atrioventricular node, raced through the bundle of His, split into the bundle branches, and finally dispersed through the Purkinje network to reach every corner of the ventricles. But the story is not yet complete. The delay at the AV node, that brief 90-millisecond pause, was more than just a mechanical necessity. It was a moment of profound coordination, carefully timed hesitation that makes the difference between life and chaos. The electrical impulse has reached the atrioventricular node, and now something remarkable happens. It pauses. For roughly 90 milliseconds, a fraction of a second that you cannot consciously perceive, the signal is held at the gateway between your atria and ventricles. This delay is not an accident. It is not a limitation of biology or a flaw in the design of your heart. It is one of the most precisely calibrated features of the entire cardiac conduction system, a built-in hesitation that makes the difference between efficient pumping and mechanical failure. To understand why this delay matters, you need to picture what is happening inside your heart at this exact moment. Your atria have just received the electrical signal from the sinoatrial node. They have depolarized, their muscle fibers have contracted, and they are now in the process of squeezing blood downward through the open valves into your ventricles. The ventricles, meanwhile, are relaxed, their walls soft and compliant, expanding passively as blood flows into them. This is the filling phase, and it is essential to your heart's function as a pump. Most of the blood that enters your ventricles does so passively, simply flowing downward from the atria due to gravity and pressure differences. But the final 20 to 30% of ventricular filling comes from the active contraction of the atria, a deliberate squeeze that tops off the ventricles and ensures they are as full as possible before they begin their own contraction. This extra volume matters. The more blood your ventricles contain at the start of contraction, the more forcefully they can squeeze, and the more blood they can eject with each beat. This relationship between filling volume and contraction strength is so fundamental to cardiac function that physiologists gave it a name, the Frank-Starling mechanism. But for this mechanism to work, for the atria to successfully complete their contraction and deliver that final bolus of blood into the ventricles, there must be time. If the ventricles receive the electrical signal at the same moment the atria did, both chambers would contract simultaneously. The valves between them would slam shut immediately, and the atria would be squeezing against closed doors, unable to push their blood forward. The result would be inefficient filling, reduced stroke volume, and a heart that cannot meet your body's demands for oxygen and nutrients. This is where the atrioventricular node comes in. Unlike the rapid conduction you saw in the atria, where the signal spread at speeds of around half a meter per second, conduction through the AV node slows dramatically to just 5 centimeters per second. This tenfold reduction in speed is achieved through the unique architecture of the AV node itself. The cells here are smaller and more densely packed than atrial muscle cells, and critically, they have fewer gap junctions connecting them to their neighbors. Gap junctions are the channels that allow electrical signals to pass directly from one cell to another. When gap junctions are abundant, signals move quickly, flowing freely from cell to cell with minimal resistance. But when gap junctions are sparse, As they are in the AV node, resistance increases and conduction slows. The AV node cells also have different electrical properties than ordinary heart muscle. Their action potentials rise more slowly, they have smaller amplitudes, and they depend more heavily on calcium currents than on the rapid sodium currents that drive conduction in other parts of the heart. The result of these features is a deliberate, carefully controlled delay. The signal enters the AV node from the atria, but instead of racing through immediately, it creeps forward slowly, taking roughly 90 milliseconds to traverse this tiny structure that is only a few millimeters across. During this time, the atria complete their contraction, the valves remain open, and blood continues to flow downward into the ventricles. Only when the atria have finished their work, when the ventricles are full and ready, does the signal finally emerge from the AV node and continue its journey downward into the ventricular conduction system. This delay is visible on an electrocardiogram, the recording of your heart's electrical activity. After the P wave, which represents atrial depolarization, there is a flat segment before the next major deflection appears. This segment is called the PR interval, and it represents the time the signal spends traveling from the sinoatrial node through the atria and then pausing at the AV node. A normal PR interval lasts between 120 and 200 milliseconds, with most of that time spent waiting in the AV node itself. The precision of this timing is extraordinary. If the delay were too short, the atria would not have enough time to finish emptying, and ventricular filling would be incomplete. If the delay were too long, the ventricles would be sitting idle, wasting time that could be spent contracting and pumping blood. The 90 millisecond delay is not arbitrary. It is the optimal balance, fine-tuned by evolution to maximize the efficiency of your heart under normal resting conditions. But the AV node does more than just create a fixed delay. It also responds dynamically to changes in your heart rate and your body's needs. When your heart rate increases during exercise or stress, the delay at the AV node becomes slightly shorter, allowing your heart to pump more frequently without sacrificing filling time. This adjustment is controlled by your autonomic nervous system, the same system that regulates the firing rate of the sinoatrial node. The sympathetic nervous system releases norepinephrine and epinephrine, hormones that enhance the flow of calcium into AV node cells, speeding up their action potentials and reducing the delay. The parasympathetic system, working through the vagus nerve, does the opposite, releasing acetylcholine that slows conduction and prolongs the delay. This autonomic control allows your heart to adapt its timing to match your activity level, ensuring efficient pumping whether you are asleep or running up a flight of stairs. But the AV node has another function, one that is just as important as creating the delay itself. It acts as a protective filter, shielding your ventricles from dangerously rapid electrical signals that might occasionally arise in the atria. This protective function depends on a unique property called decremental conduction. Decremental conduction means that the more frequently the AV node is stimulated, the slower it conducts. This might seem counterintuitive at first, but it is actually a critical safety mechanism. Imagine a situation where something goes wrong in the atria, where the electrical signals begin firing not at the normal rate of 60 to 100 times per minute, but at 200, 300, or even 400 times per minute. This can happen in conditions called atrial flutter or atrial fibrillation, where the normal coordinated depolarization of the atria breaks down into chaotic, rapid electrical activity. If every one of these rapid atrial signals were allowed to pass through to the ventricles would be driven at impossibly fast rates. At such speeds, there would be no time for proper filling, no time for the heart to function as a pump. The ventricles would flutter uselessly, unable to generate meaningful blood flow, and consciousness will be lost within seconds. The AV node prevents this catastrophe through decremental conduction. When rapid signals arrive from the atria, the AV node begins conducting them more and more slowly with each beat. Eventually, the conduction becomes so slow that some signals do not make it through at all. The AV node starts blocking beats, allowing perhaps only every second or every third atrial impulse to reach the ventricles. This filtering effect protects your ventricles from electrical chaos, maintaining a sustainable ventricular rate even when the atria are misfiring wildly. This protective mechanism is related to the AV node's refractory period, the time after each action potential during which the cells cannot be stimulated again. Every cell in your heart has a refractory period, a mandatory recovery time during which the ion channels that drive the action potential are temporarily inactivated and cannot respond to new stimuli. In the AV node, this refractory period is relatively long, and it becomes even longer when the node is being stimulated rapidly. The refractory period ensures that if a new signal arrives too soon after the previous one, it will simply be blocked. The cell has not yet recovered, the ion channels are not ready, and the signal cannot pass through. This is not a failure of the system. It is a design feature, a built-in limit that prevents the heart from being driven at rates that would be physiologically impossible to sustain. Cardiologists can observe decremental conduction in the electrophysiology lab by pacing the atria at progressively faster rates and watching what happens to conduction through the AV node. At slow pacing rates, every atrial impulse conducts through the AV node to the ventricles, though with a small delay. As the pacing rate increases, the delay at the AV node begins to lengthen. At even faster rates, the AV node starts dropping beats, conducting only every other atrial impulse or every third in predictable patterns. This behavior, known as Wenckebach periodicity, is a direct manifestation of the AV node's decremental conduction and prolonging refractory period. The exact mechanism of decremental conduction is complex and still not fully understood, but it appears to involve several factors. The sparse gap. Junctions between AV node cells mean that each cell must be individually depolarized, and when the rate of stimulation is very high, some cells simply do not have time to recover before the next impulse arrives. The calcium-dependent action potentials of AV node cells also play a role, as calcium channels take longer to recover from inactivation than the sodium channels found in other parts of the heart. There are circumstances where the AV node's conduction can become pathologically slow or blocked. This condition is called heart block, and it comes in several degrees. First-degree heart block is simply an unusually long PR interval on the electrocardiogram, indicating that conduction through the AV node is slower than normal but still functional. Second-degree heart block occurs when some atrial impulses fail to conduct to the ventricles, resulting in dropped beats. Third-degree, or complete heart block, is when no atrial impulses reach the ventricles at all, and the AV node or the tissue below it must take over as the pacemaker to keep the heart beating. Heart block can result from damage to the AV node caused by heart attacks, infections, medications, or aging. When the blood supply to the AV node is interrupted, the cells can become ischemic and lose their ability to conduct signals. Some medications, particularly those used to treat high blood pressure or abnormal heart rhythms, can also slow AV node conduction, sometimes excessively. And as the heart ages, fibrous tissue can gradually infiltrate the conduction system, replacing functional cells with scar tissue that cannot conduct electricity. When heart block becomes severe, the only treatment may be implantation of an artificial pacemaker, a device that can generate electrical impulses and stimulate the heart to beat. Modern pacemakers are remarkably sophisticated, able to sense the heart's natural rhythm and provide backup stimulation only when needed. They are a testament to medical engineering, but also a reminder of just how critical the AV node's function is to sustaining life. The contribution of atrial contraction to ventricular filling is significant, though not always in the way you might expect. When you are at rest, with your heart beating slowly and steadily, atrial contraction accounts for only about 10 to 20% of the blood that fills your ventricles. Most of the filling happens passively, simply through blood flowing downward from the pulmonary veins and vena cava through the atria and into the ventricles while they are relaxed and their valves are open. But this percentage is not fixed. When your heart rate increases, when you exercise or experience stress, there is less time for passive filling between beats. The ventricles spend less time in their relaxed state, and more of the filling burden shifts to active atrial contraction. Under these conditions, the atrial contribution can rise to as much as 40% of ventricular filling, a phenomenon cardiologists call the atrial kick. This is why the timing of the AV node delay becomes even more critical during physical activity. The faster your heart beats, the more you depend on that final squeeze from your atria to maintain adequate cardiac output. People who lose this coordinated timing, such as those who develop atrial fibrillation, where the atria quiver chaotically rather than contracting in an organized manner, often notice a significant decline in their exercise tolerance. Without the atrial kick, their ventricles do not fill as completely, stroke volume drops, and they feel short of breath with exertion. The loss of the AV node's carefully timed delay, the absence of that 90-millisecond pause, has real, tangible consequences for how the heart performs under stress. The PR interval on an electrocardiogram is a direct representation of this timing. It measures the time from the beginning of the P wave, when the atria first begin to depolarize, to the beginning of the QRS complex, when the ventricles start their own depolarization. A normal PR interval ranges from 120 to 200 milliseconds. This window is narrow but important. If the PR interval is too short, less than 120 milliseconds, it may indicate the presence of an abnormal accessory pathway that bypasses the AV node, conducting signals too rapidly and potentially causing dangerous arrhythmias. If the PR interval is too long, greater than 200 milliseconds, it suggests that conduction through the AV node is abnormally slow, a condition called first-degree heart block. Research has shown that even modest prolongations of the PR interval, increases that are still technically within the normal range but at the upper end, are associated with increased risks of developing heart failure and atrial fibrillation later in life. Every 29 millisecond increase in PR interval duration is associated with a 13% greater risk of heart failure over 10 years. A PR interval longer than 200 milliseconds carries a 46% increased risk. These associations suggest that the PR interval is not just a measurement of timing, but a marker of overall cardiac health, reflecting the integrity of the conduction system and the heart's ability to maintain coordinated function. The AV node's delay is so fundamental to cardiac function that evolution has built multiple layers of control into it. Autonomic nervous system input adjusts the delay moment by moment based on your body's needs. Hormones circulating in your bloodstream can modulate conduction speed. The intrinsic electrical properties of the AV node cells themselves provide a baseline delay, even in the absence of any external signals. And the decremental conduction property acts as a fail-safe, protecting your ventricles from dangerously rapid rates if something goes wrong upstream. This tiny cluster of cells, no bigger than a grain of rice, sitting at the junction between your atria and ventricles, is performing one of the most precisely calibrated tasks in all of human physiology. It is creating a 90-millisecond pause over and over billions of times throughout your life, with a consistency and reliability that is nothing short of remarkable. This pause is not dead time. It is not wasted. It is the moment that allows your heart to coordinate, to synchronize, to function as the efficient pump that keeps you alive. The electrical signal has now completed its pause. It has waited in the AV node for precisely the right amount of time, allowing the atria to finish their work and the ventricles to fill completely. The delay is over. The signal now emerges from the AV node and enters the bundle of His, accelerating dramatically as it begins its descent into the ventricles. What happens next will determine whether your heart can generate the powerful, coordinated contraction needed to send blood surging through your entire circulatory system. The signal emerges from the atrioventricular node, released from its 90-millisecond pause, and immediately accelerates. The moment it enters the bundle of His, the speed of conduction increases dramatically, from the slow 5 centimeters per second of the AV node to nearly 2 meters per second, a 40-fold increase. This sudden acceleration is not random. It is the result of a fundamental change in the tissue through which the signal is traveling. The bundle of His is composed of specialized cardiac muscle cells that are unlike either the atrial muscle or the ordinary ventricular muscle that surrounds them. These cells are larger, wider, and contain fewer of the contractile proteins that give heart muscle its strength. They are not built for contraction. They are built for conduction. Their membranes are packed with sodium channels that open rapidly and conduct ions with minimal resistance. Their gap junctions, the protein channels that connect one cell to the next, are abundant and large, allowing electrical signals to flow freely from cell to cell with almost no delay. This specialized conducting tissue forms a narrow corridor, a highway of sorts, that carries the electrical impulse from the AV node down into the heart. The bundle of His is roughly 20 millimeters long and 4 millimeters in diameter, a slender cord of tissue embedded in the upper part of the interventricular septum, the muscular wall that divides your left ventricle from your right. It travels mostly on the left side of the septum, running just beneath the endocardium, the smooth inner lining of your heart. This location is strategic. The interventricular septum is the central pillar of your heart's architecture, a thick wall of muscle that separates the two pumping chambers and provides structural support for the entire organ. By running through the septum, the bundle of His can reach both ventricles quickly, positioning itself to deliver the electrical signal to both chambers simultaneously. The bundle is surrounded by dense fibrous tissue, part of the cardiac skeleton, which insulates it and prevents the signal from leaking out prematurely into the surrounding ventricular muscle. The blood supply to the bundle of His comes from two sources. The upper portion receives blood from the AV nodal artery, the same vessel that supplies the atrioventricular node. The lower portion is fed by septal perforator branches of the left anterior descending artery, a major coronary vessel that runs down the front of the heart. This dual blood supply is another example of biological redundancy, a backup system that helps ensure the bundle continues to function even if one artery becomes partially blocked. The journey through the bundle of His is brief, lasting only 30 to 40 milliseconds. In that fraction of a second, the signal travels down through the septum, racing toward a critical junction point. The bundle does not remain a single pathway for long. At a location called the interventricular crest, roughly at the top of the muscular portion of the septum, the bundle of His divides. This bifurcation marks a profound moment in the electrical journey through your heart, the point where the signal splits and begins to travel separately into your left and right ventricles. The division creates two distinct pathways, the right bundle branch and the left bundle branch. These branches are not identical. They differ in size, structure, and the routes they take through the ventricular muscle. The right bundle branch is narrower and more compact, a single cord of tissue that travels down the right side of the septum before extending outward into the right ventricular wall. In many hearts, it runs through a structure called the moderator band, a muscular bridge that spans the cavity of the right ventricle and connects the septum to the anterior papillary muscle on the outer wall. The left bundle branch is larger and more complex. It carries most of the conducting fibers from the bundle of His, reflecting the fact that the left ventricle is larger and more muscular than the right. The left ventricle must generate far greater pressure because it pumps blood to your entire body, while the right ventricle only needs to push blood through your lungs, a much shorter distance requiring far less force. The left bundle branch emerges from the septum and travels down the left side, initially running superficially beneath the endocardium before diving slightly deeper into the muscle. As it descends, the left bundle branch divides again, splitting into two fascicles, the left anterior fascicle and the left posterior fascicle. These two pathways spread the electrical signal throughout the large mass of the left ventricle, ensuring that every region receives the impulse quickly and efficiently. The anterior fascicle travels toward the front and upper portion of the left ventricle, while the posterior fascicle spreads toward the back and lower regions. Both bundle branches continue their rapid descent through the septum, carrying the signal downward with remarkable speed. The conduction velocity through these pathways is approximately 2 meters per second, roughly 40 times faster than conduction through the AV node. This speed is essential because the ventricles are large chambers, and they must contract in a coordinated, synchronized manner to pump blood effectively. If different regions of the ventricles receive the signal at different times, if some areas began contracting while others were still relaxed, the result would be a chaotic, inefficient squeeze that cannot generate the pressure needed to circulate blood through your body. The bundle branches do not carry the signal all the way to the ventricular muscle themselves. Instead, as they descend through the septum and spread into the ventricular walls, they give rise to an extensive network of even more specialized conducting fibers called Purkinje fibers. These fibers, named after the Czech anatomist Jan Evangelista Perkin, who discovered them in 1839, represent the final stage of the heart's specialized conduction system. They are the ultimate delivery mechanism, the fine branches at the end of the conducting pathway that bring the electrical signal directly to the ventricular muscle cells. Purkinje fibers are remarkable cells. They are the largest cells in the heart, 2 to 3 times wider than ordinary ventricular muscle cells, and they are pale in appearance because they contain very few myofibrils, the contractile proteins that give heart muscle its ability to squeeze. These cells are not designed for contraction. They are designed purely for conduction, for transmitting electrical signals as rapidly as possible. Their membranes are densely packed with sodium channels, and their gap junctions are large and abundant, allowing ions to flow freely from cell to cell with minimal resistance. The result of this specialized architecture is extraordinary speed. Purkinje fibers conduct electrical impulses at velocities up to 4 meters per second, faster than any other tissue in the heart. This is twice as fast as conduction through the bundle branches, 8 times faster than conduction through ordinary ventricular muscle, and 80 times faster than conduction through the AV node. This speed is absolutely critical because it allows the signal to reach all parts of both ventricles almost simultaneously, ensuring that the entire ventricular muscle mass contracts in a coordinated, synchronized manner. The Purkinje network spreads like the branches of a tree, extending from the bundle branches outward across the inner walls of both ventricles. The fibers run beneath the endocardium, the smooth inner lining of the heart, forming a dense mesh that covers the inner surface of the ventricular chambers. From there, they penetrate into the thickness of the ventricular wall, reaching toward the outer surface, the epicardium. This inside-to-outside pattern of penetration is essential to the proper sequence of ventricular contraction. Ventricular depolarization follows a specific, predictable sequence. The first region to depolarize is the interventricular septum, the wall that divides the left and right ventricles. Within the septum, depolarization begins on the left side and moves toward the right. This occurs because the left bundle branch sends fibers into the septum slightly earlier than the right. Bundle branch does. The septal depolarization is rapid, taking only about 20 milliseconds, and it creates the first deflection of the QRS complex on an electrocardiogram, a small negative wave called the Q wave. After the septum depolarizes, the signal spreads to the apex, the pointed bottom tip of the heart. The Purkinje fibers carry the impulse down the length of the bundle branches to the base of the heart, then spread the signal outward through the apical muscle. This creates a wave of depolarization that moves from the apex upward toward the base, from the bottom of the ventricles toward the top. This bottom-to-top pattern is crucial because it ensures that blood is squeezed upward and out through the valves at the top of the ventricles, rather than being trapped at the bottom. At the same time that the apex is depolarizing, the signal is also spreading from the endocardium, the inner surface, toward the epicardium, the outer surface. This inside-to-outside pattern occurs because the Purkinje fibers terminate on the endocardial side, and the signal must then conduct through ordinary ventricular muscle to reach the outer layers. Instantaneous from a mechanical perspective. The entire ventricular muscle mass receives the signal to contract within roughly 80 to 100 milliseconds, less than a tenth of a second. This synchronized activation creates a wave of contraction that sweeps through the ventricles in a coordinated pattern, squeezing blood upward toward the outflow valves. The pattern of ventricular activation, from apex to base and from endocardium to epicardium, is not arbitrary. It is the most efficient sequence for ejecting blood. By depolarizing the apex first, the signal ensures that contraction begins at the bottom of the heart and moves upward. This creates a squeezing motion that pushes blood toward the aortic and pulmonary valves at the top of the ventricles. If the sequence were reversed, if the base contracted before the apex, blood would be pushed downward away from the valves, and ejection would be far less efficient. The inside-to-outside pattern of depolarization also serves a purpose. The endocardial surface, the inner lining of the ventricles, experiences higher mechanical stress during contraction because it is closer to the center of the chamber where pressure is greatest. By depolarizing first, the endocardial cells begin generating force earlier, allowing them to contribute to pressure development throughout the entire contraction. The epicardial cells on the outer surface depolarize last but still contribute to the overall force of the squeeze. The Purkinje system is so extensive that it reaches into nearly every region of the ventricular walls. The fibers branch repeatedly, becoming finer and more numerous as they extend deeper into the muscle. Eventually, the Purkinje fibers make contact with ordinary ventricular muscle cells, and at these junctions, the signal transitions from the specialized conduction system into the working myocardium. From that point forward, the signal spreads through the ventricular muscle itself, conducted from cell to cell through gap junctions just as it spread through the atria earlier in the cycle. Conduction through ordinary ventricular muscle is slower than conduction through Purkinje fibers, but it is still relatively fast, approximately half a meter per second. This allows the wave of depolarization to spread outward from each Purkinje fiber terminal into the surrounding muscle, ensuring that even regions distant from the conducting system receive the signal within a few milliseconds. The entire journey of the electrical impulse, from its origin in the sinoatrial node to the final depolarization of the last ventricular muscle cell, takes approximately 200 to 250 milliseconds. This is visible on an electrocardiogram as the sequence from the beginning of the P wave to the end of the QRS complex. In that quarter of a second, the signal has traveled through the atria, paused at the AV node, raced through the bundle of His and bundle branches, dispersed through the Purkinje network, and spread through the entire ventricular muscle mass. But the electrical signal is only the beginning of the story. Depolarization sets the stage for contraction, but it does not directly cause the muscle to squeeze. The electrical impulse must be translated into mechanical force through a complex cascade of chemical events inside each muscle cell. This process, called excitation-contraction coupling, is what transforms an electrical signal into the physical movement that pumps your blood. The signal has now reached every corner of your ventricles. The specialized conduction system has done its job, delivering the impulse with remarkable speed and precision. The pathway from the sinoatrial node through the AV node, down the bundle of His, along the bundle branches, and finally through the Purkinje network has been completed. But this electrical activation is merely the trigger. The real work, the mechanical contraction that will eject blood into your circulation, depends on what happens next inside the muscle cells themselves. You have followed the electrical impulse as it traveled through your heart, from the sinoatrial node through the atria, pausing at the atrioventricular node, racing down the bundle branches, and spreading through the Purkinje network to trigger ventricular contraction. But now it is time to return to where it all began, to the small cluster of pacemaker cells in the sinoatrial node, and to examine more closely the remarkable mechanisms that allow these cells to fire spontaneously without any external command. The pacemaker cells of the sinoatrial node are fundamentally different from every other cell in your heart. Most cardiac cells have a stable resting potential, sitting quietly at a negative voltage until something comes along to stimulate them. They wait. They rest. They require an external signal to fire. But pacemaker cells have no true resting state. The moment they finish one action potential and repolarize, they immediately begin drifting upward again, slowly and steadily increasing their voltage in a process that will inevitably lead to the next heartbeat. This automatic, self-sustaining rhythm is called automaticity, and it is the defining characteristic of pacemaker cells. These cells do not need permission. They do not wait for instructions from your brain or your nervous system. They simply fire over and over in an unbroken cycle that began before you were born and will continue until the moment you die. The mechanism behind this automaticity lies in the unique arrangement of ion channels embedded in the pacemaker cell membranes. These channels are proteins, molecular gates that span the cell membrane and control the flow of charged particles, ions, in and out of the cell. It is the movement of these ions, flowing across the membrane in carefully orchestrated patterns, that creates the electrical signals we call action potentials. The pacemaker potential, the slow upward drift in voltage that occurs between heartbeats, is driven by several different ion currents working together. The first and perhaps most important of these is a current so unusual that scientists gave it a distinctive name, the funny current, abbreviated as IF. This current is funny because it behaves in a way that defies normal expectations. Most ion channels in your body open when the membrane becomes more positive, when the cell is depolarizing. But the channels that carry the funny current do the opposite. They open when the membrane becomes more negative, when the cell is hyperpolarized. These channels are called hyperpolarization-activated cyclic nucleotide-gated channels, or HCN channels, and they are found in abundance in the membranes of sinoatrial node pacemaker cells. When a pacemaker cell finishes an action potential and repolarizes, it reaches its most negative voltage, typically around -60 to -70 millivolts. This hyperpolarization triggers the HCN channels to open. Once open, these channels allow both sodium and potassium ions to flow through them, though they are somewhat more permeable to sodium than to potassium. The critical point is that sodium carries a positive charge, and as it flows into the cell through the HCN channels, it makes the inside of the cell gradually more positive. This slow influx of positive charge is what initiates the pacemaker potential. The voltage begins to rise, gradually and steadily, climbing upward from its most negative point toward the threshold that will trigger the next action potential. The funny current flows throughout this entire upward drift, providing a constant stream of depolarizing current that pushes the membrane potential higher and higher. But the funny current is not working alone. There is a second mechanism operating in parallel, a completely different timekeeping system that scientists call the calcium clock. This mechanism involves the spontaneous release of calcium from internal stores within the pacemaker cell. Deep inside each pacemaker cell is a structure called the sarcoplasmic reticulum, a network of membrane-enclosed compartments that serve as storage sites for calcium ions. In most heart cells, calcium is released from the sarcoplasmic reticulum only when triggered by an action potential. But in pacemaker cells, something remarkable happens. The sarcoplasmic reticulum spontaneously releases small bursts of calcium even in the absence of any external signal. These events are called calcium sparks, tiny localized releases of calcium that occur rhythmically during the pacemaker potential. When calcium is released from the sarcoplasmic reticulum, it does not simply disappear. The cell must remove this calcium from the cytoplasm to prevent it from accumulating. One of the primary mechanisms for removing calcium is a protein in the cell membrane called the sodium-calcium exchanger. This protein does exactly what its name suggests. It exchanges ions across the membrane, removing one calcium ion from inside the cell and bringing in 3 sodium ions from outside. This exchange is electrically significant. One calcium ion carries 2 positive charges, but 3 sodium ions carry 3 positive charges. The net result is that 1 positive charge enters the cell for every cycle of the exchanger. When the calcium sparks occur and the sodium-calcium exchanger activates to remove the released calcium, the exchanger brings a net positive charge into the cell. This influx of positive charge contributes to the pacemaker potential, helping to drive the membrane voltage upward toward threshold. The funny current and the calcium clock work together, reinforcing one another and ensuring that the pacemaker potential proceeds smoothly and reliably. The HCN channels provide a steady depolarizing current from the outside, while the calcium clock generates rhythmic bursts of depolarizing current from the inside. Together, these two mechanisms create a robust, redundant system that is remarkably resistant to failure. As the membrane potential continues to rise, driven by both the funny current and the calcium clock, it eventually reaches a point where another set of ion channels begins to open. These are voltage-gated calcium channels, and they come in 2 types that open at different voltages, T-type channels and L-type channels. The T-type calcium channels open first, typically when the membrane potential reaches around -50 to -40 millivolts. These channels allow a small amount of calcium to enter the cell, which pushes the voltage even higher. Then, as the membrane potential continues to rise and reaches around -40 millivolts, the L-type calcium channels open. These channels open more slowly than the T-type channels, but they conduct much more calcium. The L in L-type stands for long-lasting, reflecting the fact that these channels remain open for a relatively extended period compared to other channels. When the L-type channels open, calcium floods into the cell, and this massive influx of positive charge is what drives the rapid upstroke of the action potential. The upstroke in a pacemaker cell action potential is different from the upstroke in other cardiac cells. In atrial and ventricular muscle cells, the upstroke is driven by the sudden opening of sodium channels, which creates a very rapid, steep rise in voltage. But pacemaker cells have very few sodium channels. Their upstroke depends almost entirely on calcium channels, which open more slowly. This is why the upstroke of the pacemaker action potential is slower and less steep than the upstroke of a ventricular action potential. The maximum voltage reached during a pacemaker cell action potential is also lower, typically around 0 to 10 millivolts, compared to the 20 to 40 millivolts reached in ventricular cells. But this slower, calcium-driven upstroke is perfectly suited to the pacemaker's function. Pacemaker cells are not designed to contract forcefully. They are designed to generate electrical signals that will trigger contraction in the surrounding atrial muscle. The calcium-dependent action potential is reliable, consistent, and less susceptible to the variations in sodium channel availability that can affect other cardiac cells. Once the action potential reaches its peak, the calcium channels begin to close, and the cell must repolarize, returning its membrane potential back to a negative value so the cycle can begin again. Repolarization in pacemaker cells is driven by potassium channels. These channels open in response to the depolarization of the membrane, allowing potassium ions to flow out of the cell. Potassium carries a positive charge, so when it leaves the cell, it makes the inside more negative, causing the membrane potential to fall. The potassium channels involved in pacemaker cell repolarization include several different types, each with slightly different properties. There are voltage-gated potassium channels that open rapidly in response to depolarization, and there are other potassium channels that open more slowly but remain active for longer periods. Together, these channels allow enough potassium to leave the cell to bring the membrane potential back down to around -60 to -70 millivolts. But here is the critical point: the potassium channels do not hold the membrane potential at this negative value. In most cardiac cells, there are inward rectifier potassium channels that remain open at negative voltages, maintaining a stable resting potential. But in pacemaker cells, these inward rectifier channels are less active. As soon as the membrane repolarizes to around -60 millivolts, the potassium channels begin to close, and the HCN channels begin to open again. The pacemaker potential starts immediately, and the entire cycle repeats. This is why pacemaker cells never truly rest. There is no resting phase, no stable equilibrium. The moment repolarization is complete, the pacemaker potential begins anew, driven by the funny current and the calcium clock, steadily climbing toward the threshold that will trigger the next action potential. This continuous cycling is the essence of automaticity. The rate at which the pacemaker potential rises determines how quickly the next action potential will occur, and therefore how fast your heart beats. If the pacemaker potential rises steeply, threshold is reached quickly, and the heart rate is fast. If the pacemaker potential rises more gradually, threshold takes longer to reach, and the heart rate is slower. This is how your autonomic nervous system controls your heart rate, by adjusting the slope of the pacemaker potential. The sympathetic nervous system, which becomes active during stress or exercise, releases norepinephrine and epinephrine. These hormones bind to receptors on the pacemaker cells and increase the activity of several key components of the pacemaker mechanism. They enhance the funny current by making the HCN channels more sensitive, increase calcium release from the sarcoplasmic reticulum, and boost the activity of the L-type calcium channels. All of these effects make the pacemaker potential rise more steeply, bringing the cell to threshold faster and increasing the heart rate. The parasympathetic nervous system, working through the vagus nerve, does the opposite. It releases acetylcholine, which binds to different receptors on the pacemaker cells and activates a special type of potassium channel. When these channels open, potassium flows out of the cell, making the inside more negative. This has two effects. It hyperpolarizes the membrane potential, moving it further from threshold, and it reduces the slope of the pacemaker potential by providing a constant outward flow of potassium that opposes the inward flow of sodium through the HCN channels. The result is a slower heart rate. The balance between sympathetic and parasympathetic activity changes throughout the day, adjusting your heart rate to match your body's needs. When you are asleep, parasympathetic tone dominates, and your heart rate slows to its minimum. When you wake and begin moving, sympathetic activity increases, and your heart rate rises. During intense exercise or moments of fear or excitement, sympathetic stimulation can drive your heart rate to its maximum, sometimes exceeding 180 beats per minute. But even without any autonomic input, even if all nerve signals to the heart were somehow blocked, the pacemaker cells would continue to fire. They have an intrinsic rate, a baseline rhythm built into the cells themselves, typically around 100 beats per minute. This intrinsic rate reflects the fundamental properties of the ion channels and the calcium clock, the molecular machinery that drives automaticity. The molecular machinery of the pacemaker cells is remarkably stable, but it is not immune to the effects of aging. As you grow older, the function of your sinoatrial node gradually changes. The intrinsic firing rate of the pacemaker cells decreases, the calcium clock becomes less robust, and the responsiveness to autonomic nervous system signals diminishes. Studies in aging mice have shown that the sarcoplasmic reticulum in old pacemaker cells releases less calcium, the spontaneous calcium sparks become smaller and less frequent, and the expression of key proteins involved in calcium cycling declines. These age-related changes explain why maximum heart rate decreases with age, and why older hearts have more difficulty accelerating during exercise or stress. The pacemaker reserve, the ability of the sinoatrial node to speed up when needed, becomes progressively limited. In some individuals, aging can lead to sinoatrial node dysfunction, a condition where the pacemaker fires too slowly or erratically, causing symptoms like dizziness, fatigue, or fainting. When this happens, the only treatment may be implantation of an artificial pacemaker to supplement or replace the failing natural pacemaker. But for most of your life, the sinoatrial node functions with extraordinary reliability. The pacemaker cells fire continuously, day after day, year after year, never pausing, never resting. Consider the magnitude of this achievement. If your heart beats 70 times per minute, that is over 100,000 beats per day, more than 37 million beats per year. Over an 80-year lifespan, your sinoatrial node will initiate nearly 3 billion heartbeats. No machine humans have ever built can operate continuously for 80 years without maintenance. Yet these tiny cells accomplish this feat as a matter of course. The sinoatrial node also has backup mechanisms built into its very structure. Recent research has revealed that the human SAN is not a single, homogeneous cluster of pacemaker cells, but rather a complex structure with multiple regions, each capable of generating pacemaker activity. There is a central, dominant pacemaker region, but there are also backup pacemaker sites in the head and tail regions of the node. These backup pacemakers are less sensitive to certain chemical disturbances that might disable the central pacemaker, providing redundancy that helps maintain heart rhythm even under adverse conditions. This structural and functional redundancy extends beyond the sinoatrial node itself. If the SA node were to fail completely, the atrioventricular node would take over as the pacemaker, firing at its own intrinsic rate of 40 to 60 beats per minute. If the AV node also failed, cells in the bundle of His or even the Purkinje fibers could generate spontaneous rhythms, though at even slower rates. These hierarchical backup systems ensure that even in the face of disease or injury, some rhythm continues, some electrical activity persists, keeping the heart beating and maintaining circulation. The pacemaker cells of the sinoatrial node are a testament to the elegance of biological design. They are not mechanical. They are not electronic. They are living cells, powered by the same metabolic processes that sustain all your tissues, driven by the coordinated action of proteins and ions and chemical gradients. Yet they perform their task with a precision and consistency that rivals any human-made device. The funny current, the calcium clock, the voltage-gated calcium channels, the potassium channels that drive repolarization. All of these components work together in a carefully orchestrated dance, a molecular ballet that repeats billions of times over the course of your life. Each beat is not a copy of the one before. Each is a new event, a fresh cycle of ion movements and voltage changes, yet the pattern remains remarkably consistent. And this rhythm does not exist in isolation. It is embedded in a larger context, connected to your autonomic nervous system, responsive to your body's changing needs. When you run, your heart speeds up. When you sleep, it slows down. When you feel fear or excitement, it races. When you relax deeply, it settles into a calm, steady cadence. All of this modulation, all of this adaptation happens through subtle adjustments to the pacemaker mechanism, changes in the slope of the pacemaker potential, shifts in the balance between depolarizing and repolarizing currents. The pacemaker cells have been firing since before you were conscious, since before you could think or remember. Your heart began beating around 3 weeks after conception, when you were smaller than a grain of rice, and it has continued without pause ever since. The rhythm that sustains you now is the same rhythm that has been with you for your entire existence. It is happening right now, in this moment, as you lie still and breathe quietly. The funny current is flowing. The calcium clock is ticking. The pacemaker potential is rising toward threshold. And in another fraction of a second, your sinoatrial node will fire again, sending another electrical impulse through your heart, triggering another beat, sustaining your life for one more cycle. Your heart does not beat in isolation. The pacemaker cells of the sinoatrial node, remarkable as they are, do not operate independently of your body's needs. They respond. They adapt. They adjust their firing rate moment by moment based on signals they receive from your nervous system, from hormones circulating in your blood, from the demands your tissues place upon your cardiovascular system. This constant adjustment, this continuous fine-tuning of heart rate happens without any conscious thought or effort on your part. It is controlled by a division of your nervous system called the autonomic nervous system, the part that regulates functions you do not actively think about: breathing, digestion, pupil dilation, and heart rate. The autonomic nervous system has 2 major branches that work in opposition to one another, creating a balance that allows precise control over your heart rate. The sympathetic nervous system is the accelerator, the branch that speeds things up when your body needs more oxygen and nutrients. The parasympathetic nervous system is the brake, the branch that slows things down when your body can afford to conserve energy. These 2 systems work in constant dialogue with your heart, adjusting its rhythm to match the circumstances of your life. When you are at rest, lying quietly as you are now, parasympathetic activity dominates. The primary nerve carrying parasympathetic signals to your heart is the vagus nerve, the 10th cranial nerve, which descends from your brainstem and sends branches to both the sinoatrial node and the atrioventricular node. The vagus nerve releases a chemical messenger called acetylcholine onto the pacemaker cells. Acetylcholine binds to receptors on the surface of these cells, proteins called muscarinic receptors, which then activate a signaling cascade inside the cell. When muscarinic receptors are activated, they cause several changes in the pacemaker cells that slow the heart rate. First, they open a special type of potassium channel that allows potassium ions to flow out of the cell. Potassium carries a positive charge, so when it leaves, it makes the inside of the cell more negative, hyperpolarizing the membrane potential. This hyperpolarization moves the starting point of the pacemaker potential further from threshold, meaning it will take longer for the cell to reach the voltage needed to fire an action potential. Second, acetylcholine reduces the activity of the funny current, the unusual sodium current carried by HCN channels that drives the early phase of the pacemaker potential. With less funny current flowing, the pacemaker potential rises more slowly, taking longer to reach threshold. The combined effect of these mechanisms is a reduction in heart rate. The stronger the vagal activity, the slower your heart beats. At night, when you sleep deeply, vagal tone is at its highest, and your heart rate drops to its minimum, sometimes as low as 40 to 50 beats per minute in trained athletes. This is not dangerous. It is efficient. Your body's metabolic needs are low during sleep, and there is no reason to maintain a higher heart rate than necessary. The parasympathetic system allows your heart to rest, to work at its most economical pace, conserving energy for when it will be needed. But when you wake, when you begin to move, when you stand up or climb stairs or experience stress, the balance shifts. Parasympathetic activity decreases, and sympathetic activity increases. The sympathetic nervous system sends signals through nerve fibers that also reach the sinoatrial node and other parts of the heart. These nerves release a different chemical messenger, norepinephrine, also called noradrenaline. In addition, your adrenal glands, small organs sitting atop your kidneys, release epinephrine, also known as adrenaline, into your bloodstream, where it circulates throughout your body and reaches your heart. Norepinephrine and epinephrine bind to different receptors on the pacemaker cells, proteins called beta-adrenergic receptors. When these receptors are activated, they trigger a cascade of events inside the cell that have the opposite effect of acetylcholine. They increase the activity of the funny current, making the HCN channels more sensitive and allowing more sodium to flow into the cell during the pacemaker potential. They enhance calcium release from the sarcoplasmic reticulum, strengthening the calcium clock mechanism. They increase the opening of L-type calcium channels, allowing more calcium to enter during the upstroke of the action. Potential. All of these effects combine to make the pacemaker potential rise more steeply. Threshold is reached faster. The cells fire more frequently. Your heart rate increases. During intense exercise or moments of fear or excitement, sympathetic stimulation can drive your heart rate to its maximum, sometimes exceeding 160 or even 180 beats per minute, depending on your age and fitness level. This is the autonomic nervous system at work, the invisible hand that adjusts your heart rate to match your body's needs. You do not think about it. You do not will it to happen. Yet it is happening right now, in this moment, as subtle changes in nerve activity and hormone levels fine-tune the firing rate of your pacemaker cells. The autonomic nervous system does not simply set your heart rate and leave it fixed. It continuously adjusts it, beat by beat, in response to an ever-changing array of signals from your body. This continuous adjustment creates something called heart rate variability, the natural fluctuation in the time interval between successive heartbeats. Heart rate variability is not random noise. It is a signature of health, a reflection of your autonomic nervous system's ability to respond flexibly to your body's needs. If your heart were beating with perfect mechanical regularity, like a metronome, with exactly the same interval between every beat, it would actually be a sign of dysfunction. A healthy heart shows variability, with each interval between beats differing slightly from the last. This variability exists at multiple timescales. Beat-to-beat variations of just a few milliseconds occur constantly, driven primarily by your breathing. Slower variations occur over minutes or hours, reflecting changes in your activity level, emotional state, and circadian rhythms. The most prominent source of beat-to-beat heart rate variability is your breathing, a phenomenon called respiratory sinus arrhythmia. When you inhale, your heart rate increases slightly. When you exhale, it decreases. This rhythmic variation happens because breathing affects the pressure in your chest cavity, which in turn influences the activity of your vagus nerve. During inhalation, the descent of your diaphragm decreases intrathoracic pressure, which reduces vagal output to the heart, allowing the heart rate to rise. During exhalation, intrathoracic pressure increases, enhancing vagal activity and slowing the heart. This respiratory variation in heart rate is mediated entirely by the parasympathetic nervous system. The sympathetic system responds too slowly to create beat-to-beat changes. It is the vagus nerve, with its rapid action through acetylcholine, that creates the quick adjustments in heart rate that track with your breathing. The magnitude of this respiratory variation is an indicator of parasympathetic tone, of how actively your vagus nerve is modulating your heart. Higher heart rate variability generally indicates better health and greater cardiovascular fitness. It suggests that your autonomic nervous system is functioning well, that it can respond flexibly to changes, that there is robust parasympathetic activity providing moment-to-moment fine-tuning of heart rate. Lower heart rate variability, a more rigid metronome-like heartbeat, is associated with increased risk of cardiovascular disease, heart failure, and even mortality. It suggests that the autonomic nervous system has lost some of its flexibility, that the parasympathetic brake is no longer functioning optimally. Beyond breathing, your heart rate is also continuously adjusted by another critical reflex mechanism, the baroreceptor reflex, also called the baroreflex. This is a rapid negative feedback system that helps maintain stable blood pressure. Baroreceptors are specialized stretch-sensitive neurons embedded in the walls of your aortic arch and your carotid arteries, the major vessels that carry blood away from your heart and up to your brain. These baroreceptors are constantly sensing the pressure in your arteries by detecting how much the arterial walls are being stretched. When blood pressure rises, the arterial walls stretch more, and the baroreceptors fire action potentials more rapidly. When blood pressure falls, the walls stretch less, and the baroreceptors fire more slowly. These signals travel through the vagus and glossopharyngeal nerves to a region in your brainstem called the nucleus tractus solitarius, which integrates the information and orchestrates the appropriate autonomic response. If blood pressure rises too high, the increased baroreceptor firing triggers an immediate compensatory response. The brainstem increases parasympathetic activity and decreases sympathetic activity. Acetylcholine is released to the heart, slowing the heart rate. Sympathetic stimulation to the heart is withdrawn, reducing contractility. Blood vessels throughout the body dilate as sympathetic vasoconstrictor signals are reduced. The combined effect is a rapid drop in blood pressure back toward normal levels. Conversely, if blood pressure drops too low, the decreased baroreceptor firing prompts the opposite response. Parasympathetic activity decreases and sympathetic activity increases. The heart rate rises. Contractility increases. Blood vessels constrict. Blood pressure climbs back up. This entire reflex loop can begin to respond in less than one heartbeat, in fractions of a second, making it essential for dealing with sudden changes in blood pressure that occur with everyday activities like standing up, climbing stairs, or experiencing emotional stress. The baroreflex is why you do not feel dizzy every time you stand. When you move from lying down to standing, gravity pulls blood downward into your legs, temporarily reducing the amount of blood returning to your heart and dropping your blood pressure. But the baroreceptors sense this drop instantly, and within a second or two, your heart rate has increased and your blood vessels have constricted enough to restore normal blood pressure to your brain. This happens automatically, without any conscious effort. The baroreflex, like heart rate variability, is continuously active, making small adjustments to your heart rate and vascular tone moment by moment. Even at rest, baroreceptors are firing at a baseline rate, providing ongoing feedback about blood pressure. This tonic activity allows the system to respond to both increases and decreases in pressure. The baroreflex contributes to the overall variability in your heart rate, adding another layer of dynamic control on top of the respiratory variations. Your heart rate also follows a circadian rhythm, a daily cycle that repeats roughly every 24 hours. This rhythm is not driven by your activity level or your conscious choices. It is built into your biology, controlled by a master clock in your brain called the suprachiasmatic nucleus, located deep within your hypothalamus. This internal clock coordinates countless physiological processes throughout your body, including your heart rate, adjusting them to match the natural cycle of day and night. During the daytime, when you are awake and active, your heart rate is higher. During the night, when you sleep, it drops significantly. This pattern persists even if you remain lying still all day, even if you do not change your activity level. The circadian system drives these changes by modulating the balance between sympathetic and parasympathetic activity. During sleep, parasympathetic tone increases, vagal activity dominates, and your heart slows. During waking hours, sympathetic activity rises, and your heart accelerates. Recent research has revealed something surprising about the circadian control. The sympathetic nervous system, traditionally thought of as the system that responds to stress and exercise, also plays a role in regulating resting heart rate across the day-night cycle. Sympathetic nerve activity itself follows a circadian rhythm, and this rhythmic activity influences the expression of ion channels in the pacemaker cells of the sinoatrial node. The genes that code for these channels are turned on and off at different times of day, altering the electrical properties of the pacemaker cells and adjusting the intrinsic heart rate. This means that your heart's rhythm is not just passively controlled by external nerve signals. The pacemaker cells themselves are undergoing daily changes in their molecular composition, remodeling their ion channel expression to match the time of day. Your heart is keeping its own internal time, synchronized with your body's master circadian clock. Beyond the autonomic nervous system, hormones also influence your heart rate. The adrenal glands release epinephrine into the bloodstream during stress, and this hormone circulates throughout your body, reaching your heart and stimulating the pacemaker cells in much the same way that norepinephrine from sympathetic nerves does. Thyroid hormones released by your thyroid gland also affect heart rate over longer timescales, modulating the expression of receptors and ion channels in cardiac cells. The heart itself produces hormones. Atrial natriuretic peptide, or ANP, is released by the atrial muscle cells when the atria are stretched by increased blood volume. This hormone helps regulate blood pressure by causing blood vessels to dilate and by promoting sodium and water excretion by the kidneys. Brain natriuretic peptide, or BNP, is released by the ventricles in response to stretch, and it serves similar functions. These cardiac hormones can also influence the autonomic nervous system, creating feedback loops that help maintain cardiovascular homeostasis. All of these systems, the parasympathetic nerves, the sympathetic nerves, the baroreceptors, the circadian clock, the hormones, work together in an intricate web of control, adjusting your heart rate moment by moment to match your body's ever-changing needs. When you are calm and still, as you are now, parasympathetic activity dominates, and your heart beats slowly and efficiently. When you rise and begin to move, when you encounter stress or excitement, the balance shifts, sympathetic activity increases, and your heart accelerates. This continuous adjustment happens entirely without your awareness. You do not think about speeding up your heart when you stand. You do not consciously slow it when you relax. The autonomic nervous system handles it all, monitoring your body's state through sensors embedded in your blood vessels, lungs, and muscles, integrating this information in your brainstem, and sending precisely calibrated signals to your heart's pacemaker cells. The electrical impulse that begins in the sinoatrial node, that travels through your heart and triggers each beat, is not generated in isolation. It is shaped by this invisible network of nerves and hormones, modulated by your breathing, your blood pressure, your time of day, your emotional state. The rhythm of your heart is the rhythm of your life, continuously adapting to keep you functioning, to keep your tissues supplied with oxygen, to keep you alive. The signal has been generated, modulated, and transmitted, but its work is not yet complete. The electrical impulse is only the beginning of the story of each heartbeat. What happens next, inside the muscle cells themselves, will determine whether that electrical signal translates into the powerful mechanical contraction your body needs. The electrical impulse has traveled through your heart, spreading from the sinoatrial node through the atria, pausing at the atrioventricular node, racing down the bundle branches, and dispersing through the Purkinje network to reach every region of your ventricles. The wave of depolarization has swept through the muscle cells, changing their voltage, opening ion channels, creating action potentials. But electricity alone does not pump blood. The electrical signal is merely the trigger, the command that initiates a far more complex process. What happens next, inside each muscle cell, is a cascade of chemical events so intricate and precisely coordinated that it transforms an electrical impulse into the physical force needed to squeeze your heart and propel blood through your entire body. This transformation is called excitation-contraction coupling, the process by which an electrical stimulus becomes a mechanical response. In cardiac muscle, this coupling depends on a mechanism unique to the heart, something called calcium-induced calcium release. The name describes exactly what happens. Calcium entering the cell triggers the release of even more calcium from internal stores, amplifying the signal and providing the fuel needed for contraction. The process begins with the action potential itself. As the wave of depolarization spreads across the surface of a cardiac muscle cell, it does not remain confined to the outer membrane. The cell membrane of cardiac muscle cells is not a simple, smooth surface. It has deep invaginations, narrow tunnels that penetrate from the surface deep into the interior of the cell. These tunnels are called transverse tubules, or T-tubules, and they serve a critical function. They allow the electrical signal to reach the interior of the cell almost instantaneously. When the action potential travels down into the T-tubules, it depolarizes the membrane all the way through the thickness of the muscle cell. This depolarization triggers voltage-sensitive proteins embedded in the T-tubule membrane called dihydropyridine receptors, or DHP receptors. These receptors are also L-type calcium channels, the same type of channels you encountered in the pacemaker cells, though here they serve a different purpose. When the membrane depolarizes during phase 2 of the cardiac action potential, the plateau phase, these L-type calcium channels open. Calcium ions, which are far more concentrated outside the cell than inside, flow down their concentration gradient and enter the cell through these open channels. This influx of calcium from outside the cell is the first critical step in excitation-contraction coupling. But the amount of calcium that enters through the L-type channels is not enough on its own to cause contraction. The intracellular calcium concentration rises only slightly, from around 1/10,000,000th of a mole per liter to perhaps a few 10 millionths. This increase is significant, but it is not sufficient to activate the contractile machinery. The cell needs far more calcium than this to generate force. This is where calcium-induced calcium release comes in. The T-tubules are positioned very close to another structure inside the cell called the sarcoplasmic reticulum, a network of membrane-enclosed compartments that serve as storage sites for calcium ions. The sarcoplasmic reticulum surrounds the myofibrils, the contractile units of the muscle cell, and is packed with calcium at concentrations thousands of times higher than the concentration in the cytoplasm. Embedded in the membrane of the sarcoplasmic reticulum are calcium release channels called ryanodine receptors. These receptors are sensitive to calcium. When calcium from outside the cell flows through the L-type channels in the T-tubule membrane, it reaches the ryanodine receptors on the nearby sarcoplasmic reticulum. The calcium binds to these receptors, causing them to open. When the ryanodine receptors open, calcium floods out of the sarcoplasmic reticulum into the cytoplasm. This is a massive release, far larger than the initial influx from outside the cell. The intracellular calcium concentration rises dramatically from around 1/10,000,000th of a mole per liter to 100,000th of a mole per liter, a 100-fold increase. This surge of calcium is what actually triggers contraction. The entire process happens with remarkable speed. The action potential depolarizes the membrane. The L-type channels open. Calcium enters. The ryanodine receptors open. Calcium floods out of the sarcoplasmic reticulum. All of this occurs within a few milliseconds. The electrical signal has been amplified into a chemical signal, a wave of calcium that will now activate the contractile proteins. This calcium amplification is essential to cardiac function. Without it, the small amount of calcium that enters through the L-type channels would not be enough to generate the force needed to pump blood. The sarcoplasmic reticulum serves as a reservoir, storing calcium between beats and then releasing it on demand, providing the burst of calcium needed for each contraction. The calcium that floods into the cytoplasm does not directly cause the muscle to contract. It must first interact with the contractile machinery, the molecular structures that generate force. The interior of each cardiac muscle cell is packed with myofibrils, cylindrical bundles of proteins arranged in repeating units called sarcomeres. The sarcomere is the fundamental unit of muscle contraction, and it contains two types of protein filaments, thick filaments made of myosin and thin filaments made of actin. Under a microscope, sarcomeres appear striated, with alternating light and dark bands. The dark bands are regions where thick and thin filaments overlap. The light bands are regions where only thin filaments are present. In the center of each sarcomere is a Z disc, a protein structure that anchors the thin filaments. The distance from one Z disc to the next defines the length of the sarcomere, and during contraction, this distance shortens as the filaments slide past one another. The thin filaments are not just pure actin. They also contain regulatory proteins called troponin and tropomyosin. Tropomyosin is a long, thin protein that wraps around the actin filament in a spiral pattern. When the muscle is relaxed, tropomyosin sits in a position that blocks the binding sites on actin where myosin heads can attach. This blockage prevents contraction from occurring. Troponin is a complex of 3 proteins attached to tropomyosin at regular intervals along the thin filament. One of these proteins, troponin C, is specifically designed to bind calcium. When calcium is absent, the troponin complex holds tropomyosin in its blocking position. But when calcium binds to troponin C, everything changes. The binding of calcium to troponin C causes a conformational change, a shift in the shape of the protein. This change pulls on tropomyosin, causing it to slide deeper into the groove of the actin helix. As tropomyosin moves, it exposes the binding sites on actin where myosin heads can attach. The protective barrier is removed and contraction can begin. The myosin molecules that make up the thick filaments have a distinctive shape. Each myosin molecule has a long tail and a globular head. The tails bundle together to form the backbone of the thick filament, while the heads project outward, reaching toward the thin filaments. Each myosin head has 2 important properties. It can bind to actin, and it has enzymatic activity that allows it to break down ADP, the cell's energy currency. When tropomyosin moves and exposes the binding sites on actin, the myosin heads immediately attach, forming what is called a cross-bridge. This attachment is not static. The myosin head is spring-loaded, cocked into a high-energy position by the previous hydrolysis of ATP into ADP and phosphate. When the myosin head binds to actin, it releases the phosphate, and this triggers a conformational change called the power stroke. During the power stroke, the myosin head pivots, pulling the actin filament toward the center of the sarcomere. This movement slides the thin filaments past the thick filaments, shortening the sarcomere and generating force. The myosin head then releases ADP and remains attached to actin in a state called the rigor configuration. For the cycle to continue, for the myosin head to release from actin and recock itself for another power stroke, a new molecule of ADP must bind to the myosin head. When ADP binds, it causes the myosin head to detach from actin, breaking the crossbridge. The ATP is then hydrolyzed into ADP and phosphate, and the energy released by this hydrolysis is used to recock the myosin head, moving it into the high-energy position. The head is now ready to bind to a new site further along the actin filament and execute another power stroke. This cycle repeats over and over as long as calcium remains bound to troponin C and ADP is available. The myosin heads walk along the actin filaments, pulling them toward the center of the sarcomere with each power stroke. Thousands of myosin heads in each sarcomere, millions of sarcomeres in each muscle cell, and billions of muscle cells throughout your heart all perform this molecular dance simultaneously, generating the tremendous force needed to eject blood from your ventricles. The sliding filament model, as this mechanism is called, was first proposed in the 1950s and has been confirmed by decades of research. It explains how muscle generates force at the molecular level, how chemical energy stored in ADP is converted into mechanical work. Every heartbeat, every contraction of every muscle in your body depends on this fundamental process of actin and myosin sliding past one another. In cardiac muscle, this contraction is powerful but brief. The action potential that triggered it lasts only about 200 milliseconds. The plateau phase, during which calcium enters through the L-type channels and the ryanodine receptors remain open, accounts for most of this duration. But eventually, the action potential ends, the membrane repolarizes, and the L-type channels close. Calcium stops entering the cell from outside. Once the influx of calcium ceases, the cell must actively remove calcium from the cytoplasm to end a contraction and allow relaxation to occur. Calcium does not simply disappear on its own. It must be pumped back into storage or expelled from the cell, and this requires energy. The primary mechanism for removing calcium from the cytoplasm is an ADP-dependent calcium pump located in the membrane of the sarcoplasmic reticulum. This pump, called SERCA, which stands for sarcoplasmic/endoplasmic reticulum calcium ADPase, actively transports calcium ions from the cytoplasm back into the sarcoplasmic reticulum, restoring the high calcium concentration in the storage compartment. SERCA uses the energy from ATP hydrolysis to move calcium against its concentration gradient, from a region of low concentration in the cytoplasm to a region of high concentration in the sarcoplasmic reticulum. As SERCA pumps calcium back into the sarcoplasmic reticulum, the intracellular calcium concentration falls. When the concentration drops below the threshold needed to keep calcium bound to troponin C, calcium dissociates from the troponin complex. Without calcium bound, the troponin complex returns to its original conformation, and tropomyosin slides back into its blocking position on the actin filament. The myosin binding sites are covered once again. When tropomyosin blocks the binding sites, myosin heads can no longer attach to actin. The cross bridges break as ATP binds to the myosin heads and causes them to detach. With no new cross bridges forming, the force of contraction ceases. The elastic elements in the muscle and the surrounding connective tissue pull the sarcomeres back to their resting length. The muscle relaxes. This entire cycle of contraction and relaxation is remarkably efficient, but it consumes enormous amounts of energy. Every power stroke requires one molecule of ADP. Every time SERCA pumps calcium back into the sarcoplasmic reticulum, it uses ATP. Additional energy is needed to maintain ion gradients across the cell membrane, to pump sodium out and potassium in through the sodium-potassium ATPase. Your heart beats more than 100,000 times per day, and each beat requires this massive expenditure of energy. Cardiac muscle cells are therefore packed with mitochondria, the cellular organelles responsible for producing ATP. In fact, mitochondria occupy roughly 1/3 of the volume of a cardiac muscle cell, far more than in most other cell types. These mitochondria work continuously, breaking down glucose and fatty acids through aerobic respiration, generating the ADP needed to sustain contraction. Your heart consumes oxygen at a prodigious rate, extracting more oxygen from the blood than almost any other organ. The dependence on ADP explains why the heart is so vulnerable to ischemia, to reductions in blood flow that limit oxygen delivery. Without oxygen, the mitochondria cannot produce ADP efficiently. Without ATP, the calcium pumps fail, calcium accumulates in the cytoplasm, and the muscle cannot relax properly. Without ATP, the myosin heads cannot detach from actin, and the muscle enters a state of rigor. Within minutes of complete loss of blood flow, cardiac muscle cells begin to die. But under normal circumstances, your heart has an abundant supply of oxygen-rich blood delivered through the coronary arteries. The mitochondria produce ADP continuously, the calcium pumps work reliably, and the cycle of contraction and relaxation repeats seamlessly, beat after beat, hour after hour. The process of excitation-contraction coupling, from the opening of L-type channels to the sliding of actin and myosin filaments, is one of the most thoroughly studied mechanisms in all of physiology. Yet it remains remarkable in its complexity and precision. The transformation of an electrical impulse into a mechanical force involves dozens of proteins, each with specific roles, each essential to the process. The electrical signal was the beginning, but this chemical cascade is the heart of each heartbeat. The calcium entering through voltage-gated channels, the massive release from the sarcoplasmic reticulum, the binding to troponin, the exposure of myosin binding sites on actin, the formation of cross bridges, the power strokes pulling the filaments past one another, the consumption of ADP, the pumping of calcium back into storage, the relaxation that prepares the muscle for the next contraction. All of this happens in a fraction of a second, billions of times throughout your life. And this process is happening right now, in your chest, in the walls of your ventricles. The electrical signal has just arrived, calcium is flooding into the cytoplasm, troponin is shifting, tropomyosin is moving, myosin heads are attaching to actin, power strokes are being executed, and your heart muscle is squeezing, generating the pressure that will push blood out into your aorta and pulmonary artery, sending oxygen and nutrients to every cell in your body. This is the chemistry of contraction, the molecular choreography that transforms an electrical command into the physical force that sustains your life. The contraction is complete. Your heart muscle has squeezed, the ventricles have ejected blood into your aorta and pulmonary artery, and the force that drove that contraction is now dissipating. Calcium is being pumped back into the sarcoplasmic reticulum. The cross bridges between actin and myosin are breaking, and the sarcomeres are lengthening as the muscle relaxes. But the work of each heartbeat is not yet finished. Before your heart can contract again, before the next electrical impulse can trigger the next beat, every cardiac muscle cell must reset itself electrically. The cells must restore their negative resting potential, recharge their voltage-gated channels, and prepare their membranes for the next action potential. This process is called repolarization, and it is just as essential to cardiac function as depolarization itself. Without proper repolarization, your heart cannot beat rhythmically. The cells would remain depolarized, unable to respond to the next electrical signal, unable to generate another action potential. Repolarization is the recovery phase, the restoration, the return to baseline that makes continuous, repetitive cardiac function possible. Repolarization in cardiac muscle cells is driven primarily by the movement of potassium ions. During the action potential, when the cell depolarizes, sodium and calcium ions flow into the cell, making the inside positive. To restore the negative voltage, these positive charges must be removed. The cell accomplishes this by opening potassium channels in the membrane, allowing potassium ions to flow out of the cell. Potassium is more concentrated inside cardiac cells than outside, maintained by the constant action of the sodium-potassium pump. When potassium channels open, potassium flows down its concentration gradient from inside to outside. Each potassium ion that leaves carries one positive charge with it, making the inside of the cell progressively more negative. This outward flow of positive charge is what drives repolarization. But repolarization in cardiac muscle is not a simple, instantaneous process. Unlike skeletal muscle, where repolarization happens rapidly, cardiac muscle repolarizes slowly, over a period of 200 to 300 milliseconds. This prolonged repolarization is deliberate, and it serves a critical purpose. The long action potential creates a long refractory period, a time during which the cell cannot be stimulated to fire again. This refractory period protects the heart from dangerous rapid firing and ensures that each contraction is followed by adequate time for relaxation and refilling. The cardiac action potential in ventricular muscle has 5 distinct phases, and repolarization accounts for 3 of them. After the rapid upstroke of phase 0, driven by sodium influx, the cell enters phase 1, an early, brief repolarization. During phase 1, sodium channels close rapidly, and a transient outward potassium current activates, causing a membrane potential to drop slightly. This creates the notch visible in recordings of the action potential. But then something unusual happens. The repolarization pauses. The membrane potential remains relatively stable for a prolonged period, creating a plateau that lasts 150 to 200 milliseconds. This is phase 2, the plateau phase, and it occurs because calcium channels remain open, allowing calcium to continue flowing into the cell. The inward calcium current balances the outward potassium current, and the membrane potential hovers near 0 millivolts, neither depolarizing further nor repolarizing. The plateau phase is what distinguishes cardiac action potentials from those in neurons or skeletal muscle. It prolongs the action potential dramatically, making it last more than 100 times longer than a nerve action potential. This prolonged depolarization serves several functions. It extends the time during which calcium is elevated inside the cell, allowing sustained contraction. It also creates a long absolute refractory period, during which the cell cannot fire another action potential, no matter how strong the stimulus. This prevents the heart from being tetanized, from entering sustained contraction like skeletal muscle can. Eventually, the plateau phase ends. The calcium channels begin to close, reducing calcium influx, while more and more potassium channels open, increasing potassium efflux. The balance tips, and the outward potassium current becomes dominant. The cell enters phase 3, the final rapid repolarization. During phase 3, potassium flows out of the cell through multiple types of potassium channels. There are delayed rectifier potassium channels, which activate slowly in response to depolarization and remain open during the later phases of the action potential. There are inward rectifier potassium channels, which conduct more readily as the membrane potential becomes more negative, helping to stabilize the resting potential. These channels work together to drive the membrane potential rapidly back toward its resting value of around negative 90 millivolts. As the membrane potential becomes more negative, the cell's excitability gradually returns. During the absolute refractory period, which lasts through most of phase 2 and into early phase 3, the sodium channels that drove the upstroke of the action potential are inactivated, and the cell cannot fire another action potential regardless of stimulation. But as repolarization progresses into late phase 3, some of the sodium channels begin to recover from inactivation. The cell enters the relative refractory period, during which a very strong stimulus can trigger an action potential, though it will be weaker and slower than normal. Only when repolarization is complete, when the cell has returned to phase 4 and its resting potential, are all the sodium channels fully recovered and ready to generate a normal action potential. At this point, the cell is excitable again, prepared to respond to the next electrical impulse from the conduction system. The repolarization process is visible on an electrocardiogram. After the QRS complex, which represents ventricular depolarization, there is a brief pause, and then a broad, rounded wave appears called the T wave. The T wave represents ventricular repolarization, the mass recovery of millions of ventricular muscle cells as they restore their resting potentials. The interval from the beginning of the QRS complex to the end of the T wave is called the QT interval. It measures the total duration of ventricular depolarization and repolarization. The QT interval typically lasts between 350 and 450 milliseconds, depending on heart rate. The faster the heart beats, the shorter the QT interval becomes, allowing less time for repolarization between beats. This relationship between heart rate and QT interval is so consistent that cardiologists use a corrected QT interval, called QTC, which adjusts for heart rate to allow meaningful comparisons. The QT interval and the T wave provide important information about the health of the cardiac repolarization process. Abnormalities in the T wave can indicate problems with repolarization that may predispose to dangerous arrhythmias. A prolonged QT interval suggests that repolarization is taking longer than normal, which can increase the risk of a specific type of life-threatening arrhythmia called torsades de pointes, a form of ventricular tachycardia that can degenerate into ventricular fibrillation. Long QT syndrome can be inherited, caused by mutations in genes that encode cardiac ion channels, particularly potassium channels. People with these mutations have defective potassium channels that do not open properly during repolarization, causing the action potential to be abnormally prolonged. This creates a vulnerable period during which abnormal electrical activity can trigger dangerous rhythms. Long QT syndrome can also be acquired, caused by medications that block potassium channels, by electrolyte imbalances that affect potassium or calcium levels, or by heart disease. Repolarization does not occur uniformly throughout the ventricular muscle. Different regions of the ventricles repolarize at slightly different times, a phenomenon called repolarization dispersion. This dispersion exists because different types of ventricular muscle cells have different action potential durations. Cells in the endocardium, the inner layer of the ventricle, tend to have shorter action potentials than cells in the epicardium, the outer layer. Cells in the middle layer, the midmyocardium, have the longest action potentials of all. This transmural dispersion of repolarization, the variation from inside to outside across the ventricular wall, creates what is called the repolarization gradient. Interestingly, This gradient is opposite to the depolarization sequence. During depolarization, the endocardium activates first and the epicardium last. But during repolarization, the epicardium recovers first and the endocardium last. This reversal is why the T wave on an electrocardiogram is usually upright in the same leads where the QRS complex is upright. If depolarization and repolarization occurred in the same sequence, the T wave would be inverted. The transmural differences in action potential duration arise from differences in ion channel expression. Epicardial cells express more of certain types of potassium channels, particularly a transient outward potassium current that activates during phase 1. This larger potassium current causes epicardial cells to repolarize more quickly than endocardial or midmyocardial cells. Midmyocardial cells, in contrast, have less of this potassium current and a more pronounced late sodium current, both of which prolong their action potentials. Under normal circumstances, this repolarization dispersion is well controlled and poses no danger. But when dispersion becomes excessive, when the differences in repolarization times between different regions become too large, it can create conditions that favor reentrant arrhythmias. Reentry occurs when an electrical impulse, instead of dying out after the tissue repolarizes, finds a pathway through partially recovered tissue and circles back to re-excite regions that have already repolarized. This creates a self-sustaining loop of electrical activity that can manifest as tachycardia or fibrillation. Increased repolarization dispersion is associated with many conditions that predispose to sudden cardiac death. Heart failure increases dispersion by altering ion channel expression in cardiac muscle cells. Myocardial ischemia, reduced blood flow to the heart muscle, creates regions of abnormal repolarization that can serve as substrates for reentry. Electrolyte abnormalities, particularly low potassium or low magnesium, prolong action potentials and increase dispersion. The heart has developed mechanisms to limit repolarization dispersion and maintain electrical stability. This capacity is called repolarization reserve, the heart's ability to maintain normal repolarization even when one repolarization mechanism is impaired. The heart uses multiple types of potassium channels to drive repolarization, providing redundancy. If one type of potassium channel is blocked or dysfunctional, other channels can compensate, maintaining adequate repolarization. This repolarization reserve is why many people who carry mutations in potassium channel genes remain asymptomatic most of the time. Their baseline repolarization may be slightly prolonged, but the redundancy in the system allows them to maintain relatively normal cardiac function. However, when additional stress is placed on the repolarization system, by medications that block potassium channels, by electrolyte disturbances, by fever or exercise, the reserve can be exhausted and dangerous arrhythmias can emerge. The concept of repolarization reserve helps explain why some drugs that prolong the QT interval cause arrhythmias in some people, but not others. Individuals with reduced repolarization reserve, whether due to genetic variations, underlying heart disease, or electrolyte abnormalities, are more vulnerable to drug-induced arrhythmias. Testing medications for their effects on repolarization has become a critical part of drug development, and regulatory agencies require extensive evaluation of QT interval effects before new drugs can be approved. The process of repolarization requires energy, just as depolarization and contraction do. The sodium-potassium pump, which maintains the gradients of these ions across the cell membrane, consumes approximately 1/4 of all the ADP used by cardiac muscle cells. This pump works continuously, moving 3 sodium ions out of the cell and 2 potassium ions in with each cycle, using the energy from 1 molecule of ADP. Without this pump, the gradients would gradually dissipate, and the cell would lose its ability to generate action potentials. The importance of the sodium-potassium pump becomes apparent in conditions where ATP production is impaired. During myocardial ischemia, when blood flow to the heart muscle is reduced, oxygen delivery falls, and the mitochondria cannot produce ADP efficiently. The sodium-potassium pump begins to fail, sodium accumulates inside the cells, and potassium leaks out. The resting potential becomes less negative, the cells become partially depolarized, and their excitability changes. Repolarization becomes impaired, action potential duration increases, and the risk of arrhythmias rises dramatically. Repolarization also depends on proper calcium handling. After each contraction, calcium must be removed from the cytoplasm and pumped back into the sarcoplasmic reticulum by SERCA, the calcium pump. Some calcium is also expelled from the cell by the sodium-calcium exchanger, which trades 3 sodium ions in for each calcium ion out. If calcium removal is impaired, if calcium remains elevated in the cytoplasm after contraction, it can trigger delayed after depolarizations, spontaneous depolarizing events that occur during the relaxation phase. These after depolarizations can reach threshold and trigger premature beats or more serious arrhythmias. The interdependence of depolarization, contraction, and repolarization creates a delicate balance in cardiac muscle. Each process depends on the one before it, and each must be completed properly for the next to occur normally. Depolarization triggers contraction by admitting calcium into the cell. Contraction requires the sustained elevation of calcium. Repolarization begins only when calcium channels close and calcium is removed from the cytoplasm. And the next depolarization can occur only after repolarization is complete and the ion channels have recovered. This cycle repeats with every heartbeat, billions of times throughout your life. Each cycle must be precise. Each phase must occur at the right time and for the right duration. Too much depolarization, and the cell could remain stuck in a depolarized state. Too little repolarization, and the cell would not be ready for the next beat. Too much calcium, and the muscle could enter sustained contraction. Too little calcium, and contraction would be weak. The regulation of these processes involves hundreds of proteins, ion channels, pumps, exchangers, regulatory molecules, all working in concert. Each protein must be expressed in the right amount, localized to the right place in the cell, and regulated appropriately in response to changing conditions. The genes encoding these proteins must be transcribed, the mRNA translated, the proteins folded correctly, transported to their destinations, and inserted into membranes. Any disruption in this complex system can lead to dysfunction. Yet under normal circumstances, this system works with extraordinary reliability. The heart repolarizes properly after every contraction, recovers fully between beats, and maintains its ability to respond to the next electrical signal. The QT interval remains within normal limits. The T wave appears as expected on the electrocardiogram, and the heart continues its rhythmic cycle. Repolarization is the forgotten phase of the heartbeat, less dramatic than a contraction follows, less obvious than the electrical impulse that precedes it. But it is just as essential. Without proper repolarization, the heart cannot beat continuously. Without the recovery phase, there can be no next contraction. Repolarization is the art of resetting, of returning to baseline, of preparing for what comes next. And right now, as you lie still and breathe quietly, your heart is repolarizing. The ventricles have contracted, blood has been ejected, and now millions of muscle cells are opening their potassium channels, allowing potassium to flow out, restoring their negative membrane potentials. The T wave is being inscribed on an invisible electrocardiogram, representing this mass recovery. The sodium-potassium pumps are working, the calcium is being sequestered, the ion gradients are being restored. In another fraction of a second, repolarization will be complete, the cells will be ready, and the next electrical impulse will arrive from the sinoatrial node to begin the cycle once again. Your heart does not beat at the same rate all day long. Right now, as you lie still in the quiet darkness, your heart is beating slowly, perhaps 60 or 70 times per minute, a gentle rhythm that matches your body's minimal demands. But if you were to rise suddenly, to climb a flight of stairs or break into a run, your heart rate would accelerate dramatically, perhaps doubling or even tripling within seconds. This is not a malfunction. It is adaptation, a fundamental property of your cardiovascular system that allows it to match its output to your body's ever-changing needs. The adjustment of heart rate in response to activity is one of the most important mechanisms your body uses to maintain adequate oxygen delivery to your tissues. When you are at rest, your cells consume oxygen at a relatively low rate. Your skeletal muscles are relaxed, your brain is in a quiet state, your digestive system is working at baseline levels. Under these conditions, your heart can meet your body's needs with a modest cardiac output, the volume of blood pumped per minute. Cardiac output is determined by two factors, heart rate and stroke volume, the amount of blood ejected with each beat. At rest, a typical adult heart pumps about 5 liters of blood per minute, roughly the total volume of blood in your entire circulatory system. But when you become active, everything changes. Your skeletal muscles begin consuming oxygen at rates 10 or 20 times higher than at rest. Your breathing accelerates, your body temperature rises, metabolic waste products accumulate. Your tissues need more oxygen and nutrients, and they need waste products removed more quickly. The cardiovascular system must respond, and it does so through multiple coordinated mechanisms. The first and most immediate response is an increase in heart rate. Even before you begin exercising, in the moments when you are merely thinking about moving, your heart rate begins to rise. This anticipatory increase is driven by reduced parasympathetic activity. The vagus nerve, which had been actively slowing your heart during rest, withdraws its inhibitory influence. Acetylcholine release decreases, potassium channels close, and the pacemaker potential in the sinoatrial node begins to rise more steeply. Your heart rate climbs from 60 to perhaps 70 or 80 beats per minute before you have even moved. As exercise begins, sympathetic nervous system activity increases. Nerves descending from your brain activate sympathetic pathways throughout your body. Norepinephrine is released at the sinoatrial node, binding to beta-adrenergic receptors on the pacemaker cells. Simultaneously, your adrenal glands release epinephrine into your bloodstream, and this hormone circulates to your heart, reinforcing the sympathetic signal. The combined effect is a dramatic acceleration of the pacemaker potential. The funny current intensifies, calcium release from the sarcoplasmic reticulum increases, L-type calcium channels open more readily, and threshold is reached more quickly with each cycle. Your heart rate surges upward, reaching 100, 120, 140 beats per minute or more, depending on the intensity of exercise and your level of fitness. But heart rate is not the only variable that changes during exercise. Sympathetic stimulation also increases the contractility of your heart muscle, the force with which the ventricles squeeze. Norepinephrine and epinephrine bind to beta receptors on ventricular muscle cells, enhancing calcium entry during the action potential and increasing calcium release from the sarcoplasmic reticulum. The result is a stronger contraction, a greater stroke volume, more blood ejected with each beat. At the same time, sympathetic stimulation causes blood vessels in your skin and digestive system to constrict, redirecting blood flow to your working muscles where it is needed most. These adjustments happen rapidly, within seconds of beginning exercise. Your heart rate doubles, your stroke volume increases, and your cardiac output can rise from 5 liters per minute at rest to 25 or even 30 liters per minute during maximal exercise in trained athletes. This 5-fold or 6-fold increase in blood flow is what allows your muscles to sustain high levels of activity, to extract the oxygen they need from the blood, to continue generating the ATP required for contraction. The cardiovascular response to exercise is not arbitrary or uncontrolled. It is carefully regulated by feedback mechanisms that sense your body's needs and adjust cardiac output accordingly. Chemoreceptors in your aorta and carotid arteries detect changes in blood oxygen, carbon dioxide, and pH. When you exercise, carbon dioxide production increases, and blood pH falls slightly as lactic acid accumulates. The chemoreceptors sense these changes and send signals to your brainstem, which responds by increasing sympathetic activity and further accelerating your heart. Mechanoreceptors in your muscles and joints also contribute. As your muscles contract and your joints move, these receptors send signals to the brainstem indicating that physical activity is occurring. This information is integrated with input from the chemoreceptors and baroreceptors to fine-tune the cardiovascular response. The result is a precisely calibrated increase in cardiac output that matches the demands of your working tissues. Temperature also plays a role. As your body temperature rises during exercise, your heart rate increases even if all other factors remain constant. This is partly due to a direct effect of temperature on the sinoatrial node, where warmer temperatures accelerate the pacemaker potential. It is also due to thermoregulatory responses that increase sympathetic activity and redirect blood flow to the skin for cooling. The transition from rest to exercise is not the only time your heart rate changes dramatically. Your heart rate also varies throughout the day, following a circadian pattern that reflects your body's internal clock. During the night, when you are asleep, your heart rate drops to its lowest levels, sometimes falling below 50 beats per minute in healthy, fit individuals. This nocturnal slowing is driven by high parasympathetic activity and reduced sympathetic tone. Your vagus nerve is maximally active, releasing acetylcholine that hyperpolarizes the pacemaker cells and flattens the slope of the pacemaker potential. As morning approaches, even before you wake, your heart rate begins to rise. This is not because you are moving or conscious. It is because your circadian clock, the suprachiasmatic nucleus in your hypothalamus, is orchestrating a shift in autonomic balance. Parasympathetic activity decreases, sympathetic activity increases, and your heart accelerates in preparation for the demands of the waking day. Throughout the day, your heart rate continues to fluctuate. It rises when you are active and falls when you rest. It increases during stress or excitement and decreases during calm moments. These variations are not random. They reflect the continuous adjustments your autonomic nervous system makes in response to changing conditions. Scientists have discovered that these beat-to-beat variations in heart rate, called heart rate variability, contain important information about your health. Heart rate variability measures the fluctuation in time intervals between successive heartbeats. If you were to examine a recording of your heartbeats over several minutes, you would find that the interval from one beat to the next is not constant. One interval might be 900 milliseconds, the next 920, the next 880, and so on. This variability is normal and healthy. Higher heart rate variability generally indicates better cardiovascular health and greater autonomic flexibility. It suggests that your parasympathetic nervous system is functioning robustly, that your heart can respond rapidly to changing demands, that your cardiovascular system has reserve capacity. Athletes and fit individuals typically have high heart rate variability. Their hearts show substantial beat-to-beat variation, particularly during rest and sleep. Lower heart rate variability, in contrast, is associated with increased cardiovascular risk. When the intervals between heartbeats become more regular, more metronome-like, it suggests that autonomic control has become rigid, that the parasympathetic system is less active, that the heart has lost some of its ability to adapt. Reduced heart rate variability is seen in heart failure, after heart attacks, in diabetes, and in many other conditions that impair cardiovascular function. It is also a predictor of mortality. Individuals with low heart rate variability have higher risks of death from cardiac causes. The most prominent contributor to heart rate variability is your breathing. Every time you inhale, your heart rate increases slightly. Every time you exhale, it decreases. This rhythmic variation is called respiratory sinus arrhythmia, and it is entirely normal. It occurs because breathing affects pressure in your chest cavity, which in turn modulates vagal activity. During inhalation, intrathoracic pressure decreases, vagal activity diminishes, and the heart accelerates. During exhalation, pressure increases, vagal activity strengthens, and the heart slows. You can observe this yourself. If you place your fingers on your wrist to feel your pulse and breathe slowly and deeply, you will notice that your pulse quickens as you breathe in and slows as you breathe out. This coupling between breathing and heart rate is a manifestation of healthy parasympathetic function. The stronger this respiratory variation, the better your vagal tone, and generally the healthier your cardiovascular system. Beyond breathing, many other factors contribute to heart rate variability. The baroreceptor reflex continuously adjusts heart rate in response to fluctuations in blood pressure. When you shift position, when you tense a muscle, when you experience an emotion, your blood pressure changes slightly, and your heart rate adjusts to compensate. These adjustments happen unconsciously, continuously, adding layers of variation to your heart rhythm. Your emotional state also affects heart rate variability. Stress, anxiety, and negative emotions tend to reduce variability, making the heart rhythm more rigid and regular. Relaxation, meditation, and positive emotions tend to increase variability, enhancing the natural fluctuations. This is why practices like deep breathing, meditation, and biofeedback can improve cardiovascular health. They strengthen parasympathetic function and enhance heart rate variability. Age affects heart rate variability as well. As you grow older, heart rate variability tends to decrease. This decline reflects age-related changes in autonomic function, reduced parasympathetic activity, and alterations in the structure and function of the sinoatrial node. The pacemaker cells become less responsive to autonomic signals, the ion channels change their expression patterns, and the heart's ability to vary its rate diminishes. The relationship between heart rate and physical activity is not simply a matter of cause and effect. Your heart does not passively respond to exercise. It anticipates, prepares, and adjusts in real time based on complex feedback from throughout your body. This sophisticated control system allows your cardiovascular system to meet the demands of activities ranging from sleep to intense exercise, from rest to running, from calm to excitement. When exercise ends, when you stop moving and begin to rest, your heart rate does not instantly return to its resting level. There is a recovery period during which sympathetic activity gradually decreases and parasympathetic activity gradually increases. The speed of this recovery is itself an indicator of cardiovascular fitness. Fit individuals show rapid heart rate recovery after exercise, with their heart rates dropping 50 or 60 beats per minute in the first minute after stopping. Less fit individuals show slower recovery, taking longer for their heart rates to return to baseline. This difference reflects the efficiency of parasympathetic reactivation, the ability of the vagus nerve to reassert control over the heart after sympathetic stimulation. The maximum heart rate you can achieve also changes with age. Young adults can typically reach heart rates of 180 to 200 beats per minute during maximal exercise. But maximum heart rate declines roughly 1 beat per minute per year of age, so that a 60-year-old typically has a maximum heart rate around 160 beats per minute. This decline is not due to decreased fitness. It is a consequence of age-related changes in the sinoatrial node and the heart's intrinsic pacemaker properties. The formula for estimating maximum heart rate, 220 minus your age, is widely used but is only an approximation. Individual variation is substantial, and some people can exceed their predicted maximum while others cannot reach it. What matters more than the absolute number is how efficiently your heart adjusts its rate to meet your body's demands, how quickly it accelerates when needed, and how rapidly it recovers when activity ceases. Training can improve many aspects of heart rate regulation. Endurance athletes develop lower resting heart rates, sometimes below 40 beats per minute, a phenomenon called athletic bradycardia. This is not pathological. It reflects enhanced cardiac efficiency. The athlete's heart becomes larger and stronger, capable of ejecting more blood with each beat, so fewer beats are needed to maintain adequate cardiac output at rest. The athlete also develops enhanced parasympathetic tone, with greater vagal influence on the sinoatrial node. Athletes also show greater heart rate variability than sedentary individuals, reflecting their superior autonomic function. During exercise, trained individuals can sustain higher heart rates for longer periods, and they show more efficient heart rate recovery afterward. These adaptations develop over months and years of regular training, as the cardiovascular system remodels itself in response to repeated demands. Your heart rate at this very moment reflects all of these influences. It reflects your level of physical activity, which is minimal as you lie still. It reflects the time of day, with lower rates typical during the night hours. It reflects your breathing, with slight accelerations during inhalation and slight decelerations during exhalation. It reflects your emotional state, your level of fitness, your age, and countless other factors. The rhythm of rest and activity is the rhythm of your life. Your heart accelerates when you need it to, when you move, when you climb, when you run. It slows when you can afford to conserve energy, when you rest, when you sleep, when you relax. This continuous adjustment happens automatically, orchestrated by your autonomic nervous system, guided by sensors throughout your body, executed by the pacemaker cells in your sinoatrial node. And through it all, through every acceleration and deceleration, through every beat faster or slower, the fundamental mechanism remains the same. The pacemaker cells fire, the funny current flows, calcium is released, the pacemaker potential rises to threshold, and an action potential is generated. The signal spreads through your heart, triggering contraction, pumping blood, sustaining life. The rate changes, but the process does not. Beat after beat, hour after hour, day after day, your heart adapts its rhythm to match your needs, keeping you alive through every moment of rest and every moment of activity. Your heart's electrical system is remarkably reliable, but it is not invincible. The pacemaker cells, the conduction pathways, the ion channels, the pumps and transporters, all of these components are biological, subject to damage, vulnerable to disease, capable of malfunction. Sometimes through injury, aging, genetic mutation, or illness, the rhythm that has sustained you for your entire life begins to falter. The electrical signals become irregular, too fast, too slow, or chaotic. These disturbances are called arrhythmias, and they represent moments when the heart's electrical coordination breaks down. Arrhythmias can originate from many different mechanisms. Some arise from problems with automaticity, when cells that should not be generating spontaneous electrical impulses begin to do so. Some result from abnormal conduction, when signals travel through pathways they should not use or fail to travel through pathways they should. Some emerge from reentry, when an electrical impulse circles back on itself, creating a self-sustaining loop that drives the heart at abnormal rates. Understanding these mechanisms has been one of the great achievements of cardiac electrophysiology, allowing physicians to diagnose, predict, and treat rhythm disturbances with increasing precision. The sinoatrial node, despite its reliability, can fail. Sinus node dysfunction, also called sick sinus syndrome, occurs when the primary pacemaker of the heart loses its ability to generate impulses at an appropriate rate. This can manifest as sinus bradycardia, a heart rate that is persistently too slow, sometimes dropping below 40 beats per minute even during waking hours. People with severe sinus bradycardia may feel dizzy, fatigued, or short of breath because their cardiac output is insufficient to meet their body's needs. Sinus node dysfunction can also cause sinus pauses, periods when the SA node fails to fire for several seconds. During these pauses, no electrical impulse is generated, and the heart does not beat. If the pause is long enough, blood pressure drops, blood flow to the brain decreases, and the person may lose consciousness. Fortunately, the backup pacemakers in the AV node or lower in conduction system usually take over within a few seconds, preventing prolonged asystole. But these rescue rhythms are often slower than the normal sinus rhythm, and they may not provide adequate cardiac output. The causes of sinus node dysfunction are varied. Aging is the most common factor, as the pacemaker cells gradually lose their automaticity and responsiveness over decades. The calcium clock becomes less robust, ion channel expression changes, and fibrosis infiltrates the node, replacing functional tissue with scar. Heart disease can damage the blood supply to the sinoatrial node, causing ischemic injury to the pacemaker cells. Certain medications, particularly those used to control heart rate and other conditions, can suppress sinus node function excessively. And genetic mutations in the ion channels that drive pacemaker activity can cause sinus node dysfunction even in young, otherwise healthy individuals. When sinus node dysfunction becomes severe, when the symptoms cannot be managed with medications or by removing offending drugs, the only effective treatment is often implantation of a permanent pacemaker. Modern pacemakers are sophisticated electronic devices, small enough to fit in the palm of your hand, that can sense your heart's electrical activity and deliver electrical pulses to stimulate contraction when needed. They are typically implanted just beneath the skin below the collarbone, with thin wires called leads threaded through veins into the right side of the heart. These devices can operate in multiple modes. Some pace only the ventricles, delivering impulses whenever the ventricular rate falls below a programmed threshold. Others pace both the atria and ventricles, coordinating their contraction to maintain proper timing. The most advanced pacemakers can adjust their pacing rate in response to physical activity, mimicking the natural response of the autonomic nervous system. They monitor body movement, breathing rate, or other physiological signals and increase the pacing rate during exercise, then slow it during rest. Pacemakers have transformed the lives of millions of people with sinus node dysfunction and other bradyarrhythmias, allowing them to maintain normal activity levels and avoid the debilitating symptoms of slow heart rates. The devices require periodic monitoring and battery replacement every 7 to 10 years, but they are generally reliable and well tolerated. The atrioventricular node can also malfunction, creating a different class of arrhythmias. Heart block occurs when the AV node fails to conduct electrical impulses from the atria to the ventricles properly. First-degree heart block is the mildest form, characterized by a prolonged PR interval on the electrocardiogram but with all impulses still reaching the ventricles. This condition is often asymptomatic and may require no treatment. Second-degree heart block is more serious. In this condition, some atrial impulses fail to conduct through the AV node, resulting in dropped beats. There are two types of second-degree block. Mobitz type I, also called Wenckebach block, shows progressive prolongation of the PR interval until an impulse finally fails to conduct and a QRS complex is missing. After the dropped beat, the cycle resets and the pattern repeats. Mobitz type II shows a constant PR interval with sudden, unpredictable dropped beats. Type 2 block is more dangerous because it can progress suddenly to complete heart block. Third degree, or complete heart block, is the most severe form. In this condition, no atrial impulses reach the ventricles. The atria and ventricles beat independently, each driven by their own pacemakers. The atria continue to follow the sinus node, beating at 60 to 100 times per minute, while the ventricles are driven by an escape rhythm from the AV junction or the Purkinje system, beating at 40 beats per minute or less. This dissociation between atrial and ventricular activity is called AV dissociation, and it results in severely compromised cardiac output. Complete heart block requires immediate treatment, usually with a temporary pacemaker followed by permanent pacemaker implantation. Without intervention, the slow ventricular rate cannot maintain adequate blood flow, and the person experiences severe fatigue, syncope, and risk of sudden death. While some arrhythmias result from the heart beating too slowly, others arise from excessive speed. Tachyarrhythmias are abnormally fast heart rhythms, and they can originate in either the atria or the ventricles. Atrial tachyarrhythmias include atrial flutter and atrial fibrillation, conditions where the normal coordinated contraction of the atria is replaced by rapid, disorganized electrical activity. Atrial fibrillation is the most common sustained cardiac arrhythmia, affecting millions of people worldwide. In atrial fibrillation, the atria do not contract in a coordinated manner. Instead, the atrial muscle quivers chaotically, with electrical impulses firing at rates of 300 to 600 times per minute. These rapid, disorganized signals bombard the AV node continuously, but the node's decremental conduction property protects the ventricles from being driven at impossibly fast rates. The AV node filters the signals, allowing only some impulses through, typically resulting in an irregular ventricular rhythm with rates ranging from 50 to 180 beats per minute. The loss of coordinated atrial contraction and atrial fibrillation has significant consequences. The atrial kick, the contribution of atrial contraction to ventricular filling, is lost. Stroke volume decreases, cardiac output falls, and people often feel short of breath, fatigued, or lightheaded. The irregular ventricular rhythm creates an uncomfortable sensation of palpitations, a feeling that the heart is racing or skipping beats. But the most serious risk of atrial fibrillation is stroke. When the atria do not contract properly, blood can pool and form clots in the atrial chambers, particularly in a small pouch called the left atrial appendage. If a clot breaks loose, it can travel through the circulation to the brain, blocking an artery and causing a stroke. People with atrial fibrillation have a five-fold increased risk of stroke compared to those in normal sinus rhythm. Anticoagulant medications, which prevent clot formation, are essential for most people with atrial fibrillation to reduce this risk. The causes of atrial fibrillation are multiple and often overlapping. High blood pressure, heart valve disease, heart failure, and coronary artery disease all increase the risk. Thyroid disorders, excessive alcohol consumption, sleep apnea, and obesity also contribute. In some cases, particularly in younger individuals, atrial fibrillation occurs without any identifiable structural heart disease, a condition called lone atrial fibrillation. Genetic factors play a role, and certain inherited mutations in ion channels can predispose to the arrhythmia. Treatment of atrial fibrillation focuses on three goals: controlling the ventricular rate, restoring and maintaining normal sinus rhythm when possible, and preventing strokes. Rate control is achieved with medications that slow conduction through the AV node, such as beta blockers or calcium channel blockers. Rhythm control, attempting to restore normal sinus rhythm, can be accomplished with antiarrhythmic medications or with electrical cardioversion, a procedure where a controlled electrical shock is delivered to reset the heart's rhythm. For patients with persistent or recurrent atrial fibrillation, catheter ablation can be performed, a procedure where areas of abnormal electrical activity in the atria are identified and destroyed using radio frequency energy or freezing. Ventricular arrhythmias are generally more dangerous than atrial arrhythmias because the ventricles are responsible for pumping blood to the body and lungs. Ventricular tachycardia is a rapid heart rhythm originating in the ventricles, typically defined as 3 or more consecutive ventricular beats at a rate exceeding 100 beats per minute. During ventricular tachycardia, the ventricles do not fill adequately between beats, stroke volume drops precipitously, and blood pressure falls. If sustained, ventricular tachycardia can cause loss of consciousness and can degenerate into ventricular fibrillation, a chaotic, disorganized rhythm in which the ventricles quiver uselessly and cardiac output ceases entirely. Ventricular fibrillation is a medical emergency. Without immediate treatment, death occurs within minutes. The only effective treatment is defibrillation, delivery of a high-energy electrical shock that depolarizes all the cardiac cells simultaneously, allowing the sinus node to resume control and restore a coordinated rhythm. Automated external defibrillators, or AEDs, are now widely available in public spaces, designed to allow bystanders to deliver life-saving shocks to people in cardiac arrest. The mechanisms underlying ventricular arrhythmias often involve abnormalities in repolarization. Long QT syndrome, whether inherited or acquired, creates conditions favorable for a specific type of ventricular tachycardia called torsades de pointes, French for twisting of the points, named for its characteristic appearance on an electrocardiogram. In long QT syndrome, the prolonged repolarization creates temporal dispersion, with different regions of the ventricles recovering at different times. This dispersion provides a substrate for reentry, where an electrical impulse can find a pathway through partially recovered tissue and initiate a self-sustaining arrhythmia. Many common medications can prolong the QT interval and increase the risk of torsades de pointes. Antibiotics, antipsychotics, antihistamines, and even some drugs used to treat other arrhythmias can have this effect. Electrolyte abnormalities, particularly low potassium or low magnesium, also prolong repolarization. Cardiologists must be vigilant about monitoring QT intervals in patients taking these medications and correcting electrolyte imbalances promptly. Inherited long QT syndrome results from mutations in genes encoding cardiac ion channels. More than 15 different genes have been identified, most coding for potassium or sodium channels. People with these mutations have prolonged QT intervals from birth, though symptoms may not appear until adolescence or adulthood. Triggers for arrhythmias include exercise, emotional stress, or sudden loud noises, depending on which gene is affected. Treatment includes beta blockers to reduce sympathetic stimulation and, in high-risk individuals, implantable cardioverter defibrillators that can detect and terminate life-threatening arrhythmias automatically. The heart possesses remarkable resilience in the face of electrical disturbances. Even when the primary pacemaker fails, backup systems activate. Even when one conduction pathway is blocked, alternative routes may exist. The redundancy built into the cardiac electrical system reflects millions of years of evolution, selection for reliability in a system where failure means death. The hierarchical organization of pacemaker cells exemplifies this resilience. If the sinoatrial node fails, the AV node can assume pacemaker function, though at a slower rate. If the AV node fails, cells in the bundle of His or Purkinje fibers can generate escape rhythms, though slower still. Each level of the hierarchy provides a safety net, a backup that maintains some rhythm even when higher centers malfunction. Recent research has revealed even more sophisticated backup mechanisms within the sinoatrial node itself. The node contains multiple pacemaker regions with different properties and different vulnerabilities. If one region is disabled by drugs or disease, another can take over, maintaining automaticity even under adverse conditions. This structural and functional redundancy within the primary pacemaker provides an additional layer of protection against bradycardia. Medicine has developed increasingly sophisticated tools for managing arrhythmias. Beyond pacemakers and defibrillators, catheter ablation techniques allow physicians to map the heart's electrical activity in exquisite detail, identify abnormal pathways or ectopic foci, and selectively destroy them. Three-dimensional electroanatomic mapping systems create detailed electrical maps of the heart chambers, showing where signals originate, how they propagate, and where they become disorganized. Antiarrhythmic medications have also evolved. Modern drugs target specific ion channels with increasing precision, suppressing abnormal automaticity or prolonging refractoriness in strategic locations to prevent re-entry. While all antiarrhythmic drugs carry risks, including the potential to cause arrhythmias themselves, a phenomenon called proarrhythmia, careful selection and monitoring allow many patients to maintain normal sinus rhythm. Genetic testing has opened new avenues for identifying people at risk for inherited arrhythmia syndromes before symptoms appear. Families with histories of sudden cardiac death can be screened for mutations in cardiac ion channel genes. Individuals carrying high-risk mutations can be monitored closely and treated preventively, potentially averting tragedies. Yet despite all these advances, arrhythmias remain a significant cause of morbidity and mortality. Sudden cardiac death, most often caused by ventricular fibrillation, claims hundreds of thousands of lives each year. Atrial fibrillation contributes to heart failure and stroke in millions. Heart block requires pacemaker implantation in hundreds of thousands of people annually. The vulnerability of the heart's electrical system reminds us that biology, for all its elegance and sophistication, is fragile. The ion channels can malfunction, the pacemaker cells can age and fail, conduction pathways can be damaged. A single mutation in a single gene can disrupt the delicate balance of currents that drives the pacemaker potential. A moment of ischemia can alter ion gradients and create conditions for re-entry. The same mechanisms that allow adaptability and responsiveness also create vulnerabilities. But the heart is also resilient. It has backup systems, redundant pathways, compensatory mechanisms. When one component fails, others often compensate. The repolarization reserve allows normal function even when one type of potassium channel is defective. The multiple pacemaker sites ensure that some rhythm continues even when a primary pacemaker fails. And when the heart's own resilience is insufficient, when the electrical system falters beyond its ability to self-correct, medicine can intervene. Pacemakers can replace failed natural pacemakers. Defibrillators can restore coordinated rhythm from chaos. Ablation can eliminate sources of abnormal signals. Medications can stabilize membranes and prevent dangerous arrhythmias. The rhythm that sustains your life is both robust and vulnerable, reliable yet delicate. It has been beating since before you were conscious, and in most people, it will continue without interruption until the very end. But it depends on countless molecular mechanisms functioning correctly, on genes being expressed properly, on proteins being assembled and localized accurately, on ions flowing through the right channels at the right times. When these mechanisms fail, when the rhythm falters, the consequences can be profound. Yet even in failure, Even when arrhythmias emerge, the heart often finds a way to continue. A backup pacemaker fires, an escape rhythm emerges, compensation occurs. And when natural compensation is insufficient, modern medicine offers tools that would have seemed miraculous just decades ago. The rhythm may falter, but it need not fail completely. Understanding the mechanisms of cardiac electrophysiology has given physicians the knowledge to restore rhythm when it is lost, to prevent arrhythmias before they occur, and to support hearts that can no longer maintain their rhythm alone. Your heart did not always beat the way it does now. There was a time before you could think or remember, before you were even recognizable as human, when your heart was just beginning to form. Around 3 weeks after conception, when the embryo that would become you was smaller than a grain of rice, a group of cells began to organize themselves into a primitive tube. This tube, the first structure that could be called heart, began to pulse. It had no chambers yet, no valves, no organized conduction system. But it beat. This early heart was driven by spontaneous electrical activity in cells that had differentiated into primitive pacemaker cells. These cells fired rhythmically, contracting the tubular heart and pushing fluid through the developing circulatory system. The rate was fast, around 160 beats per minute, much faster than your adult heart. This rapid rhythm was necessary because the embryonic heart was inefficient, and high rates compensated for low stroke volumes. As development progressed, the heart tube began to fold and loop, transforming from a simple cylinder into a structure with recognizable chambers. The atria formed, then the ventricles, and the septum grew to divide the chambers into left and right sides. The valves developed from endocardial cushions, tissue that would eventually become the thin flaps that prevent backward flow of blood. And the electrical conduction system began to take shape. The sinoatrial node emerged as the dominant pacemaker early in cardiac development. Specialized pacemaker cells clustered in the right atrium, establishing the rhythm that would persist for the rest of your life. The atrioventricular node formed at the junction between atria and ventricles, creating the critical delay that coordinates atrial and ventricular contraction. The bundle of his grew down the interventricular septum, branching into left and right bundle branches, and the Purkinje network spread throughout the ventricular walls. By the time you were born, your heart had been beating for approximately 36 weeks, totaling more than 30 million beats before you took your first breath. It had pumped blood through your developing body, delivering oxygen and nutrients to growing tissues, removing waste products, maintaining the circulation that allowed you to develop from a few cells into a complete human being. At birth, your heart underwent dramatic changes. Before birth, your lungs were not functioning, and your circulation included shunts that allowed blood to bypass the lungs. The moment you took your first breath, your lungs expanded, resistance in the pulmonary circulation dropped, and these shunts closed. Your heart suddenly had to pump blood through your lungs for oxygenation, a task it would perform for the rest of your life. Your newborn heart beat at a rate of around 120 to 160 beats per minute, twice as fast as an adult heart. This rapid rate was normal and necessary. Infants have higher metabolic rates than adults, requiring more oxygen delivery per kilogram of body weight. Their hearts are smaller, their stroke volumes lower, so higher rates compensate to maintain adequate cardiac output. As you grew through childhood, your heart grew with you. The muscle cells enlarged, the chambers expanded, the valves grew larger. Your heart rate gradually slowed as your heart became more efficient. By school age, your resting heart rate had dropped to around 80 to 100 beats per minute. By adolescence, it approached adult values of 60 to 100 beats per minute. The electrical properties of your heart also matured during childhood. The ion channels in the pacemaker cells changed their expression patterns, the conduction velocity through the specialized pathways increased, and the action potential duration shortened. The autonomic nervous system developed its control over heart rate, with parasympathetic tone increasing and contributing to the gradual slowing of the resting heart rate. Throughout childhood and into young adulthood, heart functioned at peak efficiency. The pacemaker cells fired with robust automaticity, the conduction system transmitted signals rapidly and reliably, and the muscle contracted forcefully. Your heart could accelerate to maximal rates exceeding 200 beats per minute during intense exercise, then recover quickly afterward. Your heart rate variability was high, reflecting healthy autonomic function and cardiovascular reserve. But your heart did not remain static. From early adulthood onward, subtle changes began to occur. Changes so gradual you would never notice them from day to day or even year to year, but cumulative over decades. The aging process affected every component of your cardiovascular system, from the pacemaker cells to the muscle fibers to the valves and blood vessels. The sinoatrial node began to show signs of aging first. The number of pacemaker cells gradually declined, replaced by fibrous tissue and fat. By age 70, the sinoatrial node may contain only 10% of the pacemaker cells present in youth. The remaining cells showed altered function, with reduced automaticity and decreased responsiveness to autonomic signals. The ion channels that drive the pacemaker potential change their expression and function with age. The funny current became less robust, the calcium clock mechanism weakened, and the L-type calcium channel showed reduced activity. These molecular changes translated into a slower intrinsic heart rate. The heart's baseline firing rate, the rate it would beat without any autonomic input, declined from around 100 beats per minute in young adults to 70 or 80 beats per minute in older individuals. The autonomic nervous system's control over heart rate also diminished with age. Parasympathetic function declined, with reduced vagal tone and less pronounced respiratory sinus arrhythmia. Sympathetic function became less responsive, with reduced beta-adrenergic. Receptor sensitivity and decreased ability to accelerate heart rate during stress or exercise. The result was reduced heart rate variability, the natural beat-to-beat fluctuations that indicate healthy autonomic function. Maximum heart rate declined predictably with age, dropping approximately 1 beat per minute per year. A 20-year-old might reach 200 beats per minute during maximal exercise, while a 70-year-old would reach only 150. This decline was not due to decreased fitness or motivation, but to fundamental changes in the pacemaker cell's ability to fire rapidly. The conduction system aged as well. The internodal pathways, the bundle of His, the bundle branches, and the Purkinje network all showed progressive fibrosis, calcification, and cell loss over decades. Conduction velocity slowed, the PR interval lengthened slightly, and the QRS duration increased. These changes were usually subtle and did not cause symptoms, but they increased the risk of conduction abnormalities and heart block. The cardiac muscle itself underwent age-related changes. The number of muscle cells decreased, and the remaining cells enlarged to compensate. Fibrosis increased, with collagen depositing between muscle fibers and reducing the heart's compliance. The ventricles became stiffer, less able to relax and fill during diastole. Calcium handling became less efficient, with reduced circa activity and impaired calcium release from the sarcoplasmic reticulum. These structural and functional changes were not disease. They were normal aging, occurring to some degree in everyone who lived long enough. But they created vulnerabilities. The aging heart was less able to tolerate additional stresses, ischemia, hypertension, valve disease, arrhythmias that might have been manageable in youth. Yet despite these changes, most hearts continue to function adequately throughout a normal lifespan. The heart's remarkable reserve capacity meant that even with reduced efficiency, even with fewer pacemaker cells and slower conduction, the heart could still maintain adequate cardiac output at rest and during moderate activity. Many people reach their 80s or 90s with hearts that, while aged, still beat reliably every day. The total number of beats your heart would produce over a lifetime was staggering. At an average rate of 70 beats per minute, your heart beat just over 100,000 times per day. That was more than 37 million beats per year. Over 70 years, your heart would beat nearly 2.5 billion times. Over 80 years, almost 3 billion. No machine humans had ever built could operate continuously for 80 years without maintenance. Yet your heart accomplished this as a matter of biological routine. Consider what those billions of beats represented. Each beat required the sinoatrial node to generate an action potential, the signal to spread through the conduction system, calcium to flood into muscle cells, actin and myosin filaments to slide past one another, ADP to be consumed, calcium to be pumped back into storage, and repolarization to reset the system for the next cycle. Each beat required precise coordination of billions of cells, trillions of protein molecules, countless ion channels, pumps, and transporters all working in concert. The heart that beat in your chest now was both the same heart you were born with and a completely different organ. The cells had turned over, proteins had been synthesized and degraded, mitochondria had divided and been recycled. The structure had been remodeled in response to growth, exercise, and aging. Yet the fundamental rhythm persisted, the same pattern of electrical activation, the same sequence of depolarization and repolarization, contraction and relaxation. The heart was a living organ, constantly adapting, constantly responding to the demands placed upon it. It grew stronger with exercise training, developing increased muscle mass and improved efficiency. It adapted to pregnancy, to illness, to changes in altitude and temperature. It compensated for injuries, routing signals around damaged tissue when necessary. It was remodeled in response to chronic demands, changing its shape and function to match the work required of it. The heart's journey through life was not one of steady decline, but of continuous adaptation. Even in aging, even as some functions diminished, others compensated. The heart learned, in a sense, to work with what it had, to maintain function despite changing circumstances. This adaptability was one of the most remarkable features of cardiac biology. Exercise and lifestyle choices profoundly influence the aging trajectory of the heart. Regular physical activity maintained cardiac efficiency, preserved autonomic function, and delayed many age-related changes. Athletes in their 60s and 70s showed heart rate variability and cardiac function comparable to sedentary individuals decades younger. The heart responded to training at any age, demonstrating that biological aging was not entirely predetermined but could be modified by how you lived your life. Nutrition, sleep, stress management, and social connections all affected cardiac health across the lifespan. Chronic stress reduced heart rate variability and increased cardiovascular risk. Poor sleep disrupted circadian rhythms and autonomic balance. Social isolation and loneliness were associated with increased rates of heart disease and mortality. The heart was not isolated from the rest of your experience. It was embedded in and responsive to every aspect of your life. Medical advances had transformed what it meant to age with a heart. Conditions that would have been fatal decades ago were now manageable. Pacemakers replaced failing sinoatrial nodes. Medications controlled arrhythmias and supported heart function. Surgical procedures repaired or replaced damaged valves. Coronary interventions restored blood flow to ischemic muscle. The heart's journey through life was now supported by technologies that extended both lifespan and quality of life. Yet technology could not replace the wonder of natural cardiac function. The sinoatrial node, that tiny cluster of cells no bigger than a grain of rice, continued to fire spontaneously, reliably, without conscious input or external power source. The funny current flowed, the calcium clock ticked, the pacemaker potential rose to threshold, and action potentials were generated. The signals spread through the conduction system with remarkable speed and precision. Calcium flooded into muscle cells, triggered contraction, and was efficiently removed. Repolarization restored the resting potential, preparing for the next beat. This process, repeated billions of times, was the rhythm of your existence. Your heart began beating before you had thoughts, before you had memories, before you were conscious of your own being. It would continue beating through every experience of your life, every joy, every sorrow, every moment of excitement, and every moment of peace. It beat while you slept, while you dreamed, while you woke to each new day. It accelerated when you were afraid or excited, slowed when you were calm or resting. It adapted to your activities, your emotions, your body's ever-changing needs. The heart's journey was your journey. From the first spontaneous contractions in the embryonic heart tube to the steady rhythm in your chest right now, your heart had been with you through every moment. It had grown as you grew, adapted as you changed, aged as you aged. It had sustained you through illness and health, through activity and rest, through every phase and transition of your life. And it would continue. Tomorrow, next week, next year, your heart would still be beating. The pacemaker cells would still be firing. The conduction system would still be transmitting signals. The muscle would still be contracting. The rhythm would persist, as it had persisted since before you were born, carrying you forward through time, sustaining your life with every beat. This lifelong journey of the heart, from its earliest embryonic contractions to its final beats, represented one of the most fundamental continuities of human existence. Your heart was the one organ that had been working without pause since the beginning of your life. Every other part of you rested periodically. Your muscles relaxed, your brain slept, digestive system paused between meals. But your heart never stopped. It was the engine of your existence, the metronome of your mortality, the steady rhythm that defined the boundaries of your life. As you lie here now, in the quiet darkness, your heart continues its ancient rhythm. The story of how it keeps beating is the story of molecular machinery, electrical signals, chemical cascades, and biological coordination of extraordinary complexity and precision. But it is also a simpler story, a story of persistence, reliability, and the fundamental drive of living tissue to maintain the patterns that sustain life. There is one more truth to understand about this rhythm that has accompanied you from your beginning. You have traveled through the architecture of your heart, following the electrical impulse from its origin in the sinoatrial node through every pathway and mechanism that makes each heartbeat possible. You have seen how specialized pacemaker cells fire spontaneously, how the funny current flows and the calcium clock ticks, how signals spread through conducting pathways with remarkable speed and precision. You have witnessed calcium flooding into muscle cells, triggering the sliding of actin and myosin filaments that generates force. You have watched as repolarization resets the system, as autonomic nerves adjust the rhythm, as the heart adapts to rest and activity, to youth and age. But now it is time to return to the simplest truth of all. Right now, at this very moment, as you lie still in the darkness, your heart is beating. It is not a memory. It is not a concept. It is happening. Somewhere deep in the right atrium of your heart, a cluster of pacemaker cells no bigger than a grain of rice is firing electrical impulses. The funny current is flowing through HCN channels. Sodium ions are entering the cells. The pacemaker potential is rising toward threshold. Calcium is being released from the sarcoplasmic reticulum in rhythmic sparks. The sodium-calcium exchanger is bringing positive charge into the cells, and the voltage is climbing steadily upward. In another fraction of a second, the membrane potential will reach threshold. L-type calcium channels will open, calcium will flood in, and an action potential will be generated. That electrical signal will spread outward through your right atrium, carried from cell to cell through gap junctions, radiating across the atrial wall like ripples on water. It will travel through Bachmann's bundle to your left atrium, ensuring both chambers contract together. It will race down the internodal pathways toward the atrioventricular node. At the AV node, the signal will pause. For 90 milliseconds, it will wait, held at the gateway between your atria and ventricles, allowing your atria time to finish contracting and push that final bolus of blood downward into your ventricles. Then the signal will emerge from the node and accelerate dramatically, racing down the bundle of his, splitting into the bundle branches, dispersing through the Purkinje network to reach every corner of your ventricles. The wave of depolarization will sweep through your ventricular muscle. L-type calcium channels will open in the T-tubules. Calcium will enter from outside, and this calcium will trigger the ryanodine receptors to release a massive flood of calcium from the sarcoplasmic reticulum. The calcium will bind to troponin C. Tropomyosin will move. Myosin binding sites on actin will be exposed, and crossbridges will form. The myosin heads will execute their power strokes, pulling the actin filaments toward the center of each sarcomere, shortening the muscle, generating force. Your ventricles will squeeze. Blood will be ejected through the aortic and pulmonary valves, surging out into your circulation to deliver oxygen and nutrients to every cell in your body. Then repolarization will begin. Calcium channels will close, potassium channels will open, potassium will flow out of your cells, and the membrane potential will return to its resting value. Circa will pump calcium back into the sarcoplasmic reticulum. The sodium-potassium pump will restore ion gradients, and your heart muscle will relax. All of this, the firing of the pacemaker cells, the spread through the conduction system, the calcium release, the contraction, the repolarization, all of this is happening right now. It is not theoretical. It is not abstract. It is as real as the air you breathe, as present as the sensation of your body resting on the bed beneath you. And in another second, it will happen again, and again, and again. This rhythm has been with you since before you were born. Your heart began beating around 3 weeks after conception, when you were smaller than a grain of rice, when you had no brain, no thoughts, no consciousness. That first primitive heart tube pulse spontaneously, driven by cells that had differentiated into the earliest pacemakers. Through all the months of development, through the formation of chambers and valves and conduction pathways, through your birth and the dramatic changes that came with your first breath, through every day of your childhood and every year of your life, your heart has continued beating. It has beaten more than 100,000 times every day, more than 37 million times every year. If you are 40 years old, your heart has beaten nearly 1.5 billion times. If you are 60, more than 2 billion. If you are 80, approaching 3 billion. Each of those billions of beats require the same intricate choreography you have learned about: pacemaker cells firing, signals conducting, calcium flowing, filaments sliding, repolarization resetting. The reliability of this rhythm is extraordinary when you truly consider it. Your heart has no off switch, no rest period, no time when it can pause for maintenance or repair. The pacemaker cells fire continuously. The conduction system transmits signals without interruption. The muscle contracts and relaxes in an endless cycle. While you sleep, while you dream, while your conscious mind drifts through rest, your heart continues its work. While you are distracted, while you are focused on other things, while you are unaware of its presence, it beats on. This is perhaps the most remarkable thing about your heart. It sustains you without requiring your attention. You do not have to remember to make it beat. You do not have to consciously regulate its rate or coordinate its chambers. You do not have to think about opening ion channels or releasing calcium or pumping ions back across membranes. All of this happens automatically, orchestrated by biological mechanisms that have been refined over hundreds of millions of years of evolution. The science behind each heartbeat is almost impossibly complex. The pacemaker potential alone involves multiple ion currents working in concert: the funny current, the calcium clock, the T-type and L-type calcium channels, the potassium channels that drive repolarization. Each current depends on specific proteins embedded in cell membranes, proteins with precise structures that allow them to sense voltage changes, bind specific ions, open and close at exact moments. These proteins are encoded by genes, transcribed into RNA, translated into amino acid sequences, folded into three-dimensional structures, transported to the right locations in the cell, and assembled into functional channels. The excitation-contraction coupling that transforms electrical signals into mechanical force involves even more layers of complexity. The T-tubules that carry depolarization deep into the cell, the L-type calcium channels that emit calcium from outside, the ryanodine receptors that release calcium from internal stores, the troponin and tropomyosin that regulate access to myosin binding sites, the myosin heads that walk along actin filaments powered by ADP, the Circa pump that recycles calcium, the sodium-potassium pump that maintains ion gradients. Each of these components is essential, and each is a marvel of molecular engineering. Yet all of this complexity works seamlessly billions of times throughout your life. The proteins fold correctly, the channels function properly, the pumps maintain gradients, the signals propagate, the muscle contracts. Occasionally, things go wrong. Ion channels malfunction, conduction pathways fail, pacemakers slow, but the redundancy built into the system often allows compensation. Backup pacemakers take over. Alternative pathways conduct signals. Repolarization reserve maintains stability. The heart is also remarkably adaptive. It responds to your autonomic nervous system, accelerating when you need more cardiac output and slowing when you can afford to conserve energy. It adjusts beat by beat to your breathing, increasing slightly with inhalation and decreasing with exhalation. It follows circadian rhythms, slowing during sleep and accelerating during waking hours. It adapts over longer timescales to training, to aging, to the changing demands of your life. The heart you have now is not the same heart you were born with. The proteins have been replaced countless times. The mitochondria have divided and been recycled. The ion channels have been synthesized anew. The structure has remodeled in response to growth and exercise and age. Yet the fundamental pattern persists, the same rhythm, the same sequence of events, the same coordination of electrical and mechanical activity that has sustained you from your beginning. There is something deeply moving about this continuity. Your heart connects your present moment to every moment that came before. The rhythm beating in your chest now is the same rhythm that beat when you were a child, when you were an infant, when you were in the womb. It is the one constant thread running through your entire existence, the one process that has never stopped, the one rhythm that has accompanied you through every experience, every change, every transition of your life. And it will continue tomorrow. When you wake, your heart will still be beating. When you rise and move through your day, it will accelerate to match your activity. When you rest in the evening, it will slow. When you return to sleep, it will settle into its lowest rates, allowing your body to recover and restore itself. Through all of this, the pacemaker cells will keep firing, the signals will keep conducting, the muscle will keep contracting. The science you have learned tonight is not separate from the experience of being alive. It is the mechanism of that experience. Every thought you have, every movement you make, every sensation you feel depends on your heart continuing to beat, continuing to pump blood, continuing to deliver oxygen to your brain and your muscles and every other tissue in your body. The electrical signals, the ion channels, the calcium release, the sliding filaments, all of these molecular events are what make your consciousness possible. There is wonder in understanding this. The more you know about how your heart works, the more remarkable it becomes. It would be easy to take the heartbeat for granted, to assume that something so constant, so reliable, must be simple. But the opposite is true. Each heartbeat is a triumph of biological coordination, a moment when billions of protein molecules work together with exquisite precision to accomplish something essential. And yet, despite all this complexity, the heartbeat has a quality of simplicity and inevitability. It just happens. The pacemaker cells fire because that is what they are designed to do. The ion channels open and close because voltage changes trigger conformational changes in their protein structures. Calcium enters cells because it flows down its concentration gradient. Myosin binds to actin because ATP provides the energy for the conformational change. Each step follows from the one before with a kind of molecular logic, a causality built into the physics and chemistry of living systems. You can rest in this knowledge. Your heart does not need your awareness to continue beating. It does not require your attention or your worry. Right now, as you settle deeper into relaxation, as your breathing slows and your body becomes heavy with approaching sleep, your heart is managing itself. Your parasympathetic nervous system is becoming more active, releasing acetylcholine at the sinoatrial node, slowing the pacemaker potential, bringing your heart right down to its resting level. This is happening automatically, without any effort on your part. The rhythm will continue through your sleep. While you dream, while you cycle through different stages of rest, your heart will keep beating. It will slow during deep sleep, reaching its lowest rates of the night, then accelerates slightly during REM sleep when your brain becomes active. In the hours before dawn, even before you wake, it will begin to speed up gradually, preparing your body for the coming day. And when you open your eyes tomorrow morning, your heart will have carried you through the night, beating steadily, reliably, sustaining your life while you were completely unaware. This is the gift your heart gives you, the freedom to live without thinking about survival. Because your heart beats on its own, you can focus on other things. You can think, feel, create, connect with others, pursue meaning and purpose. You can lie here now and simply be resting in the knowledge that the fundamental machinery of your existence is functioning, maintaining you, keeping you alive. The beat goes on. It has always gone on, and it will continue. The sinoatrial node fires, the signal spreads, calcium flows, muscle contracts, repolarization resets, and the cycle begins again. This rhythm is older than your consciousness, deeper than your thoughts, more fundamental than your identity. It is the pulse of your existence, the tempo of your life, the quiet background rhythm that makes everything else possible. And as you lie here now, feeling your body relax, feeling yourself drifting towards sleep, you can be aware of this rhythm if you choose. You can feel your heartbeat, sense the gentle pulse in your chest, notice the steady cadence that has been with you always. Or you can simply rest in the knowledge that it is happening, that your heart is beating right now, that the pacemaker cells are firing and the signals are spreading and the muscle is contracting, all without your conscious involvement. The science is beautiful because it is real. The mechanisms you have learned about tonight are not abstractions. They are happening in your body at this very moment. The funny current is flowing, the calcium clock is ticking, ion channels are opening and closing, proteins are changing shape, energy is being transformed. All of this invisible molecular choreography is occurring right now in the darkness of your chest, sustaining you, keeping you alive. Let this knowledge bring you peace. Your heart knows what to do. It has been doing this since before you were born. It will continue doing this while you sleep. You can trust the rhythm. You can rest. The beat goes on, now and always. This is the end of your journey.